Well, good morning, everybody. Thanks for coming. I'm Paul Anez with Anez Consulting. We got a quaint group today, and that's for a reason. We only invited certain people that are really we know are interested in kind of moving to the next level. Well, I mean, all farmers are, but you guys are specifically interested in improving soil health. The ways we can get to the next level with tillage and that whole mix. So thank you for coming. I think we have a smaller group, so we'll try to keep this more casual. Ask questions. If you have a question, just pause and we'll get you a microphone. We have two of them here we can pass around. So just some housekeeping. We have a porta potty back behind the bin over there, so it's a little walk, but not too far. Yeah, so the reason we're here, we put on a meeting in Little Falls this winter with a big group, and thought we'd follow up with that. I got an email from Jim this I think in January or something like that, Jim, and kind of offering to do something similar out in the field, and we took him up on it. And we thought it'd be good to partner up with Jim with Dr. Jerry Hatfield as well. They kind of work together as a team really well, and these two guys here, in my opinion anyway, I think are two of the best guys in the United States at least that know what they're talking about when it comes to tillage, to soil biology, to water, percolation, infiltration, how you get carbon into the soil, and how to make that whole system work. So it's a pleasure to have these two guys here. We had a lot of knowledge and wisdom, and so we're grateful to have them here. As far as our company, our team is, we know some things, but we're humble enough to know that we don't have all the answers, that's for sure. And so we'll do whatever it takes to lean on other people, other farmers, other people in the industry. I mean, my mentor, Del Glanzer, some of you know, he'd go far as to say, we'll even go to the competition if we have to. So, so yeah, we're excited to have you here. I think there's a lot to learn. We're going to start off with Dr. Jerry Hatfield. He's going to be speaking first, and then we'll move into Jim Martindale. And then after that, I think as part of Jim's talk, we're going to move into the field. We have a section of this field right here where last fall they ran the CurseBuster. So we got some things we can look at in the root pit that are going to be pretty interesting, I think. So yeah, we'll start off with... and also we got refreshments here. Feel free to get up at any time and help yourself. So we'll start off with Dr. Jerry Hatfield. Jerry, you've been retired since 2020, I think, and so you're probably working harder now than you have back then, I suppose. So he just told me when we were talking in the field, he's working on a big project with camelina. We just had some farmers asking about that, so that's interesting. You can bend his ear on that if he's got some time. So, yeah, the thing about Dr. Jerry Hatfield, he's been in the industry a long time, working with a lot of different industries and people, but very humble man, very passionate about agriculture, very, very passionate about helping farmers, and so excited to have him here. So, thanks for being here.
I got that one? All right. Whenever he introduces me like that, it's always like, he just must be old. That's yeah. We're gonna talk about soil health in a little bit today, and then we'll follow it up. Jim and I will actually follow it up, and when we get you down in the root pit out there at the end to draw some of these pieces together, but. The question that and this is all dialogue because you're not going to be able to see some of the pieces that are on the screen here and everything else, but the question you have to ask is relative to soil health is why care? Why do you care about soil health? And then the other piece of that puzzle that goes with that is this dynamic of really what limits your crop production. Is it water? Is it air? Is it nutrients? Pest? Whether they be insects, diseases, weeds, or is it management? And management really kind of fits it back in saying, I don't know really what limits crop production. And when you look at it and say, the most important factor that we need to overcome is more water, more nutrients. We always think that the cure to every problem is adding something else to the system. Isn't that right? We always think if we have a shortage that if there's a limitation into that crop, what do we have to add to it? And in reality, we need to think about agriculture from this perspective. And agriculture from this perspective of extractive agriculture is that we use the soil to grow the plant. In regenerative agriculture, we're using plants to grow the soil. And it really is about the overall process. Because as we begin to look at this system, it's really about how efficiently we capture energy. And day like today, we're about 60-70% of full sunlight right now. There's a little bit of haze out there. All that wildfire smoke is still causing problems across the Midwest. But in reality, what agriculture is an energy transfer process. We're taking energy from the sun, we're putting it through the leaf in terms of photosynthesis. We're taking that photosynthesis and we're growing that plant. As you'll see later, we're also growing roots. We're putting sugar from that system. And all we are in the agricultural process is that we are in the sugar capturing and redistribution process. How many people had breakfast this morning? Everybody, right? Why'd you have breakfast this morning? You were hungry. You need energy in here. If we want that plant to grow, we've got to capture energy and put it in there. If we want our microbes to grow in the soil, we've got to put energy into it. It really is the overall process of taking energy and putting it into the system and saying, what can we do with that energy and make it change in a way in which we can begin to work with this system from an entirely different perspective. If we take this corn crop throughout the whole growing season, we're going to capture megajoules, megacalories of energy. About 40% of that energy that gets captured by that leaf goes into the soil. That's a phenomenal amount of sugar that actually gets deposited below the root surface. And you wonder why certain soils don't respond in the way in which we think they're going to respond. We still got that same growing plant growing out there. We've got that sugar going into it, but why aren't they responding? Why aren't they changing? And part of it is that there are the limiting factors, water being one of them. But the most important limiting factor is actually oxygen. You have to breathe in order to work. When you breathe, you give off CO2. The same thing in terms of the microbial process is that everything below our feet that's a biological process takes oxygen in, gives CO2 back out. The more sugar we give it, the higher the respiration rate, or the higher that activity goes on. But if oxygen becomes a limiting factor, because we can't exchange it down through the profile, what happens? What happens if you put a plastic bag over your head? You can't breathe, you die pretty quickly. That's why you've got all those warnings on every plastic bag that's out there. Do not put this on your head. Bad things will happen. We need a warning on our soils as well as do not limit oxygen exchange because bad things will happen. And it's the most limiting factor, but we never talk about it. How many times have you heard somebody talk about oxygen in the soil? Rarely. How many times have you heard people talk about CO2 in the soil? A little more often. because everybody's enamored with carbon sequestration and carbon dynamics and carbon markets and things like that. So carbon is a big piece of the puzzle that's out there. But in reality, oxygen is the overall process that we really need to think about. The same process occurs as we think about this whole soil health principles and we think about reducing tillage, adding cover crops, changing crop diversity, adding livestock into that system from the carbon that comes out of that livestock is that the basis for all of our energy is the carbon part of the process. It's not nitrogen, it's not water, it's actually carbon. Carbon is the energy currency of all of this thing that we talk about. So we're in the whole process. Now we've got that plant growing out there. We're taking energy and we're putting it into that soil. What do we do with it in terms of feeding that microbial system? As we feed the microbial system, we begin to see things happen within the soil. We see things happen in the soil as we begin to change aggregate size, because all of that carbon that comes off in terms of sugars is then transported and transformed into all sorts of things. That glue that you see, that mycorrhizae that's fed along the surface, will actually show you some roots with that. Out of all of that soil that are on there, that soil that's clinging onto that root is nothing more than the fact that sugar is sticky. You think about it from that standpoint, it sticks along there. We got all those different things, that we got fungi, we got all these different things. So it's about how much energy we capture and how can we convert that energy into pieces that we take to work with. We can change aggregates and all of this, but if you think about these soil health principles, the regenerative ag, reducing tillage, keeping the crop covered, keeping that residue on the surface, keeping the diversity out there, all of those have some aspect of energy with them. And we'll start by just looking at the first piece of this puzzle. First piece of this puzzle is really that's an instantaneous change when we begin to implement some of these practices. And the instantaneous change that goes with that is the water dynamic. As soon as you add that crop out there, or you add a cover crop out there, or you add residue out there, when raindrop hits that crop, all the momentum from that raindrop is absorbed by that crop and it slowly moves down to the soil. So we can increase our infiltration rates. Once we can increase our infiltration rates, then we can get that water into the soil. The more we make that system resilient to all of this, we do that piece of the puzzle so we get more water in there. As we begin to change that residue layer and that crop cover out there, we change the whole water evaporation rate, so we're no longer evaporating water back to the atmosphere. We're keeping it in that soil so we can do something with it. And then we make that soil cooler as well. If we take and look at this system from just changing the water dynamics, that occurs within days. The next phase that occurs within this system, it came to the realization when it started looking at some of the dynamics of this, as we change the water dynamic, that occurs within hours and days. The next piece that occurs within weeks is the carbon change. We see changes in the carbon storage within that. We change in the carbon cycling and all the different process. And then in kind of a month schedule, so you got days, weeks, and months, is we change the nitrogen piece of this cycling. We change all of these different pieces, not simultaneously, because everybody thinks that when we change soil health, everything occurs at the same time, it occurs at the same rate, but in reality, there's a water change, and there's a carbon change, and then there's a nitrogen change. And we see that over and over again, and people now begin to realize that it's a series of overlapping cycles.
Jerry, can you explain a little bit about that nitrogen change?
Sure. The nitrogen change that goes with that is we change the microbial activity. What we change is mineralization rates. We change the whole dynamic of how that microbe is cycling the system and everything else. Paul asked me to talk a little bit about carbon nitrogen. We did a cool study that only insane people would do, but is that, In a corn soybean system with a cover crop and no cover crop, and this was actually some of the work that we did with Mitchell Hora and some of his staff over there, is that for 5 years we collected weekly Haney test from spring until after maturity on fall across 3 reps, corn soybean system, cover crop, no cover crop. One of the things that we saw in that is that there wasn't much of an effect of the cover crop on the corn, but a major effect of the cover crop on soybeans. You can say, well, why didn't the cover crops work? Well, cover crops only work if you give them enough time to capture energy to put energy back into the soil. Because in the corn system, even though it was planted green, it was terminated when the cover crop was only about a foot tall. So we really didn't capture much energy to put it back in and made a change in the soil. But when we took the soybean crop and let the cover crop get up to about 4 to 6 feet tall, then we begin to see all sorts of changes. We improved aggregates. We see all the different things. We improved respiration rate in all of this. It occurred in the soybean part of the overall system, and we see a difference in terms of cycling of the nitrogen that goes on within the system. We saw it much more in the soybean part of the rotation. But the thing as we went through 5 years is that we did see changes in soil health, but we also saw changes in the whole carbon-nitrogen ratios. When we had the no cover crop, the carbon-nitrogen ratio was actually higher, and when we kept putting the cover crop into the system, is that the carbon-nitrogen really became into balance because we were cycling the nitrogen at a much different rate within the system. We see all the different dynamics that are going on. So the soil becomes into balance. The soil begins to work on this. That's the effect of the microbial system within this. And so when you really begin to look at soil health, you got to really begin to understand that what's going on in water, what's going on with carbon, what's going on with nitrogen. We saw all sorts of other things with different nutrients out there, but everybody's always concerned about the nitrogen dynamics and first and foremost. But we saw changes in the respiration rate, as you would expect. Higher respiration rate, higher soil health scores in all of this. Again, because the effect of adding that additional energy into that system. We did another analysis because I spend a lot of time working in eastern Colorado right now. And the question there is, how do you take advantage of the limited water that's available, but a very harsh climate? A harsh climate because we'll get soil temperatures that are in excess of 150 to 160 degrees at the surface. So the question was asked, what if we would just change row spacing? What if you went from 30-inch rows to 10-inch rows? So I simulated what would happen in all of this. And what you find is that as you begin to narrow the row spacing and you increase that shade on that soil surface, is that you dramatically change the soil temperature, you dramatically change the rate at which water is evaporated. And at the end of the season, what we actually had was we were capturing almost twice as much energy going into that soil with the 10-inch row as we were with the 30-inch row. Same thing we see in cover crops here. As we add that cover crop, all we're doing is increasing the amount of time in which we're capturing solar radiation, putting it into sugar that we move it into that soil. And so we see a dramatic effect of this. Cover crops are nothing more than a way in which we extend the capability of taking and capturing the sunlight and put it in there. It is an energy-driven process. It's just like putting money into a bank account. If you want to have a big retirement account, what do you have to do? You have to keep putting money into it, not take it out. All your financial advisors will tell you to diversify your portfolio if you really want to build it over time. Same thing happens in the soil. If we want that soil to grow for us, What do you got to do? You got to put energy into it. You got to diversify it, and you got to continually put energy into it. You just can't put energy into a selected part of the season. And so all we're doing with a cover crop is expanding the amount of time in which we can capture that energy. And you really look at this system from an entirely different perspective. So the question now becomes, how do we overcome what limits crop production? So now it's audience participation time. What's the most limiting factor in your crop production system? Anybody? Water. Why? Okay, you can't control water. Okay. What else? Anything else? Water is the primary limitation? If we solve water problem, we solve every problem? Photosynthesis? What about photosynthesis? Why is it limiting? Okay. The whole process I just talked about. So if water is the primary limitation, how do you have to manage your system to get more water into it? Okay, got to increase the infiltration rate, got to keep the storage. You can reduce the evaporation rate coming out so that we add more into that and all the other pieces that go with that. So we can begin to look at this system from an entirely different perspective. Water is probably the primary limitation. It's actually, if we look across the Midwest, water availability accounts for about 50 to 60% of the limitation in crop yields across the Midwest. Some of it due to uneven distribution of rainfall during the growing season, too much rainfall at times. There's a statistic that we found across the Midwest is that the more it rains in April and May, the lower the average county yield. We delay planting, we drowned out spots, we got all these other problems that go with it. Excuse me. The more it rains in July and August, the higher the average county yield. Makes sense as well. Thank you, Paul. That rolls off, catch it. Everybody needs a straight line. So those two primary things, too much water in the spring, too much water in the summer, give you different responses in all of this. When we begin to improve soil health, we begin to see that we can break those correlations. We can plant on time. We've got the oxygen into the soil. We got greater water storage capacity late in the growing season when we need it. It tides us over from all these different pieces that go on. So water can be managed, even in the Midwest, but it has to be managed from managing the soil perspective. And once we do that, then we can begin to see all these different changes that go on. The other piece of this is if water's the primary one, what's the second limiting factor in production? Oxygen. Gotta have oxygen in the soil. A lot of this in terms of an efficient root system and everything else. If we don't have an efficient root system, what's that corn look like? Or what's the soybeans look like? Yeah, it's a little bit like that. A little dwarfed. I mean, the system needs to be looked at as a system. We tend to want it separated into all these different pieces, but in reality, soil health has forced us to start looking and saying, what are all the interacting factors that go on? Once you can take care of water and you can take care of oxygen, a lot of the other pieces begin to disappear. We see a lot more cycling of nutrients going into the season. Where we've seen, as we've improved our soils, is that we maintain that healthy plant all the way through the growing season. And just a little bit of dynamics of all this is that in a corn plant out there, and you see that ear at one level, all the photosynthesate that is being captured above that ear is going into the ear. Photosynthesis from the leaves on down are actually continuing to supply the roots. And so there's a differential process in all this. So we can begin to see those. So maintaining that green leaf area all the way and up and down that plant does pay advantages in all this. We don't grow any more roots, but we do keep those roots active. And active roots facilitate nutrient uptake. You see all these different pieces that go on. So you got oxygen, you got water, you got nutrients. Anything else limiting that? Bugs, diseases, weeds. There's a myriad of things that influence our production, right? We need to understand them as a system as well. So we're going to open it up for questions, because some of them will spur Jim's talk as well, but because I kind of know where he's headed. What questions do you have about soil health? Everything is, and all the soil health TikToks, YouTubes, and everything else are all self-explanatory out there? I could just hold mine over.
Okay.
I was just wondering, how do you get a cover crop? We've tried cover crops, and they never seem to be able to grow long enough. We've even tried flying it on, and so how do you maintain a cover crop long enough to make a benefit with it?
Yeah, I mean, cover crop management, I think, is still one of the major challenges we have out here, particularly in the upper Midwest. How do you get it planted? How do you get that so that you can get efficient capture of solar radiation during the fall? Another little piece that I found, just to put it in a different perspective for you, is I did a calculation of how much solar radiation was available from the time of black layer on corn until first frost, which we would stop growth on plants, and then from last frost until that corn plant began to emerge in the spring. And actually, and across the Midwest, is we have about a third of our total solar radiation for the year available in those times of the year in which we could actually grow a plant. So the question is, how do we get that plant out there to capture all that light that's available to us? So flying it on is often not as advantageous because we're flying it onto dry soil. Because here's the other little problem we have across the Midwest. We have more potential of dry soil surfaces in the fall than we do wet soil surfaces. So we're putting it out there onto a dry soil surface, expecting that little seed that's laying on top of a hot, dry soil to actually take root. You don't see a whole lot of plants growing in this yard out here, do you? There's seeds laying out there, but just it's not going to grow. And so the question is, how do we begin to break this cycle? Do we go to a shorter season crop so that you have more opportunity to put the cover crop in? How do you trade that piece of the puzzle? Do you sacrifice a little bit of productivity to be able to capture more radiation or more solar radiation? I've had this wild idea that What if we had cover crops that had a seed coating on it that we could plant in the spring, and then they wouldn't germinate until the temperatures got to a certain point in the fall? You could sow them in there, have a piece of that. All it takes is a chemist, because we've done the things the other way that have had seed coatings on that didn't that degraded when the temperatures got above a certain point. Why can't we do the opposite than that and look at it from that perspective? I think we have to have more imagination of how we think about our cover crop seed. The other question is, how do you terminate it? A lot of people are now planting green and then terminating after that. That gives a lot of trafficability across the system. Although I have a producer in eastern Kansas that hasn't terminated his rye quite well enough, and so he's had a volunteer rye crop that's now causing all sorts of problems. And so it becomes the art of how we terminate all of this. And more importantly, how do you terminate it without using a lot of chemicals that might have an impact on the biological system within the soil as well? So we need a self-destructing cover crop. Just grow it up and it'll just die automatically, you know. Sounds like oats. You know, I mean, there's all sorts of things, and so I think our biggest challenge is, how do we start looking at cover crops from a different perspective, and how do we manage it, or even going to a system that puts a third crop in a rotation that overwinters and grows, and not call it a cover crop. That becomes a different piece of the puzzle as well, in looking at that dynamic of the system. I'll admit those are probably our biggest challenges. How do we manage that crop that's intermediate to our two cash crops to make them work effectively? Seeding, getting them established, getting enough growth that it really has a positive impact, getting rid of it so it's not having a negative impact the next spring in terms of the competition. So we just need to train some weeds that would work for us from a different perspective in all of this. Okay.
Yeah, about the same question he had was just, we just do oats so it dies off over winter,
Yep.
and we don't get any growth in the spring. And you were talking how you almost need that growth in the spring to pull the nutrients up to get them back in the soil. So, I mean, is there still has to be some amount of benefit on planting something that dies over winter, depending on how much growth you get in the fall or not. But that's kind of the challenge that we have. We don't really want to come back out with another chemical in the spring if we wouldn't have to.
If we go to oats, in terms of letting that, and then get that our other crop in as quick as possible in the spring, because we would be able to plant on time and everything else, is to still capture that solar radiation and capture the energy out there. It's a matter of how much growth can we get in that oat, and the impact of all this. It really becomes a balance of the system. A lot of the Canadian producers have gone to oats because it does overwinter, allows them to get in, and they've actually seen some pretty remarkable changes in their soil health from that perspective. So I don't think anything's off the table. It's a matter of how do we begin to look at this in a system and saying, what can we take advantage of, and what do we understand, and what's the best way for a particular operation in all of this. And it may be more advantageous to go to oats and then be able to go to the earlier planning of what other crop that we wanted to capture that light, and maybe going back and even thinking about narrowing that row spacing down so that you capture light a lot quicker. Because if you went from a 30-inch row to a 15-inch row on corn, you'd capture, in the first 30 days, you'd capture about twice as much sunlight. And shade that out from that perspective. So I think there's a lot of things we need to be thinking about just on row spacing to look at that piece of the puzzle. Mark?
Yeah, Jerry,
Oh, but
when we start talking about increasing oxygen, when we start talking about increasing in oxygen and carbon cycling, are we improving our weed control and the efficacy of our herbicides?
Yeah, I think we do have an impact on efficacy of the herbicides, but I think the other piece that we have when we think about that process of improving that soil structure and everything else is that we're also changing the whole weed seed dynamic within that. Where do you see weeds go in a field? Anaerobic areas. What else?
Compacted.
Compacted areas and what's that? Sunlight. Skips, right? Anytime that we have a skip, that's where we see you got the drowned out spots, you got the aerobic conditions, you got poor parts of that field, you've got sunlight and all of this. Keeping that cover on there changes that whole weed seed dynamic and all of this. And I think this is one of the pieces of the puzzle as well, is that I have a high school classmate that keeps asking me, how much carbon do weeds put into the soil? Well, weeds can put as much carbon into the soil as do that economic crop. They're photosynthesizing just as well. You know, they've got the same root structure and everything else. If we would try to commercialize water hemp, Palmer amaranth, they'd probably die on us. You know, yeah, we don't have a good track record of trying to domesticate a lot of plants. I think if we would try to domesticate some of our weeds, they'd get rid of them. So, you know, you think about it from that perspective. But all of this, in terms of the overall process, is looking at the system and saying, why do these problems occur? Why do weeds occur? Because if we begin to change this system out there in terms of the soil properties, water, oxygen, all of this, we've seen changes in it. But a lot of it is just keeping the sunlight off that so that weed seed doesn't germinate. You can cycle a lot of things within that system. Other questions?
Jerry, if we're really struggling to get cover crops to work for whatever reason, can't we pump more carbon and sugar and into the soil by just growing a great crop? You know, by improving the soil compaction and all that, and growing a more or making sure your crop is the best shape it can be.
Yeah, I mean, let's think about this. How much energy do we capture if we grow a 320 bushel corn crop? You know, we're going to have a 10, 12-foot plant out there. We're going to capture a lot of sunlight. We're going to put it back in there. If you think about 300 bushels of corn standing out there, take that 40% of that sugar, that put it in the soil, we've got a tremendous impact of it. But it's a matter of how do we manage it afterwards so that we keep that in there. Because one of the biggest culprits that we have is how rapidly do we lose our carbon that we put back into the soil. One tillage operation will take and move that back out and run aggressive tillage operation. And I drive back and forth up and down I-35, and I'm always amazed as how much black soil there is in the fall going on. And I'll give you this statistic and all of this because we've actually measured CO2 levels in the atmosphere with some instruments that allow us to actually do all these different things. But one of the things that we see after corn harvest, and one of the instruments that we have, is that CO2 levels will go to about 400, 410 parts per million. That's where the atmospheric concentration is right now. Until everybody begins to till. And then the CO2 concentrations go up to about 600, 620 parts per million. And if you back calculate what it takes to get that level in the atmosphere, is that we've taken and put all the carbon that we captured all growing season back in the atmosphere in about a 2-week period in which everybody does their aggressive tillage. So if we reduce our tillage intensity and change the timing of that tillage. I'm not saying we completely eliminate, we just change the intensity of that, is that we can have a tremendous impact on how we capture carbon and keep carbon within the soil. Mark?
Is there any testing on maybe the biomarkers that's associated with carbon? Because if I have a giant ragweed where Paul's standing versus that corn, is the carbon the same? We know the quantity's different, but is the carbon the same? And is that giant ragweed actually giving us the carbon that's needed for certain microbes?
There is a difference in exudates to come out of it. We do see a difference in the exudates coming out of it. I haven't biomarkered exudates coming out of giant ragweed yet.
How about differences in varieties? I mean,
There are slight differences in varieties because we know that when we go to, from an experiment that we did where we actually captured the exudates coming out in the soil volume by actually measuring with very small tubes that allowed us to capture right next to roots, is that when we begin to diversify the rotation and add different crops to that in terms of a cover crop mix, we actually saw a change in the quality of that exudate. Viscosity, types of sugars that were there, all the different sugar content. So I'm sure there's a lot of difference between plant species. We just have not gotten to that yet. There's now all of this effort going on in terms of looking at biomarkers coming out. We just haven't got to the point of now getting to that segregation level of saying, here's what different corn varieties are doing, this is what different soybean varieties, this is what different weed species are doing. Because a lot of these tests, they're fairly expensive to run, but they're getting more and more routine in all of this. So there's been a lot of work being done on looking at DNA and RNA and all the other things that go on. We're running a lot with just PLFA tests right now on this, and we do see that as we change the system out there, we do see changes in terms of the whole PLFA. We're just trying to interpret really what all of that means because we don't have much of a baseline of really what's going on. So the whole soil biology process is really very fascinating. The really sad truth is we probably only know about 5% of the soil microbiome compared to the 95% we don't know. Jim says we only know 2%, but I was being generous to 5%. It's still a pretty big difference between knowing and not knowing. Let's put it that way.
Just the quick description on what you mean by extradate. I just kind of learned that lately myself, but that would maybe be helpful to some people.
Oh, sure. Think about exudate as nothing more than all the slime that's coming out of the roots. It is basically just a sugar compound that's exuded out of the root surface. And you dig down in the roots and everything else, and you find out that there is actually a coating around them it's a sugar-based compound. What we were actually measuring is just when our whole experiment was how much sugar content there was, and you can just begin to see that. And so it's just a natural process that's occurring that comes out, and it's a symbiotic relationship over time. You think about this. Here's roots. They've got microbes around them. They need to be fed, and so plants have geared themselves in a way of saying, I'm going to take care of things that help me, and so that's what the exudates are coming out, and it's a sugar-based compound. It's nothing more than carbohydrate.
And Jerry,
Yeah.
and I Jim's gonna expound on that in his talk with rhizophagia and that as well. Right. But I've heard like anywhere from 15 to 30% is kind of a typical plant percentage of the photosynthesis percentage of the sugars that's pumping back through the roots to the soil. But if you have I've also heard that if you have a healthier crop, you're gonna have a higher percentage of those sugars being pumped back through the roots to feed the biology.
Yeah, it can go as high as 40% of coming out. But think about it this way, is that here's the solar radiation curve for a day. Here's the photosynthetic curve under a nice day. But if we stress that plant a little bit in mid-afternoon and we don't have that photosynthate rate going on, what we see is that if we limit photosynthesis, we limit the amount of sugar going into that system. And so the healthier the plant, the more sugar we can capture, the more sugar we can put back in. Excuse me. More putting in there. So it really behooves us to keep that plant as healthy as possible because that will optimize sugar generation. It optimizes sugar deposits into that soil as well. And anytime we stress that plant, we see a lag in photosynthesis. Get a little bit of water stress, and that photosynthetic rate will go from optimum to maybe 50% of optimum very quickly. And if we're stressing that top part of that plant, we're going to limit the amount of translocation that goes down to those roots. We just need to have more detailed studies on a lot of these dynamics, and these are tough studies to do. It takes a lot to get those exudates out of the soil. It takes some very specialized equipment to do that.
The sugars change between vegetative and
Well, sugar.
I get it's.
Oh.
I might be nuts. It seems like my soybeans, I can have no weeds out there, right through vegetative, and then as soon as it switches over to reproduction, I seem to get weeds. Does it change where the sugars are going?
I don't know if it changes where the sugars are going. It probably is looking at opportunistic weeds that take advantage of different temperatures that are going on. Different life cycles of weed systems that work with that. So what weeds are occurring, because they could be more warm season weeds that now have an advantageous microclimate for them to express themselves. All right.
Yeah, Jerry, I don't know if you want to talk a little more about like what is an ideal carbon and nitrogen ratio to shoot for, and like Cody, we pulled some samples here. I think they're on that 10-1, and
Yep.
this is a field that the Czechs have been doing an amazing job of reducing tillage and a lot of things, and so we're still struggling to get there.
The ideal carbon nitrogen ratio is somewhere in that 8 to 10-1. If you look at the sweet spots for microbial activity, it's somewhere in that range. And if you look at a lot of the different systems, when we start getting above that, we see that that microbe has a struggle. Getting it down in that range, a lot of energy, because carbon is the energy source, nitrogen is the nutrient source. And that 10-1 in that range is probably where we need to be keeping it to have the maximum response out there. I keep explaining to people that there's a lot of things that happen in this whole system. And when we did the cover crop over that 10-year period, is that we were getting it down somewhere in that 8 to 10, but there's a lot of variation that we saw. But the average was just in that spot. And when we had the no cover crop, we were up about 12 or 15. So we could change it over time with all of that. All right. You ready, Jim?
Not as good as I'll be.
And then we'll move into the field, so now you can bring your shovels. I know. First you have to dig the pit, and then we...
Well, thanks, Jerry. So next up, we'll bring Jim Martindale. So I never even knew who Jim was until a couple of years ago when I had talked to Mark Dodd in the back, just looking for some like fish product to use, foliar feeding and in-furrow and that kind of thing. And Mark said, you need to talk to Jim. And so gave Jim a call, and it turned into a hour conversation. But Jim's brings a unique knowledge. Just like a lot of you farmers, you're good at a lot of things. You got to fix equipment, you got to know nutrition, you got to know soils and everything. Jim understands the mechanics. He's owner of Soil Regeneration Unlimited, and as they manufacture the CurseBuster tillage tool. So he really understands engineering and mechanics, but he also understands, plant nutrition, soil biology, soil function, and he also has a good understanding of plant of nutrition, cattle nutrition, and that kind of thing. So he has a unique perspective and understands that whole system. So Jim's going to kind of expound a little more on what Jerry was talking about. So thanks, Jim.
2006, the word carbon has been demonized as kind of the problem of the planet. And of course, most of the time it's talking about CO2 and fossil fuel kind of burning and things like this. It turns out, though, biologically, carbon is actually the most important substrate that we can speak of if we're going to talk about life. So what the heck is carbon? Carbon is basically the communication network for the energy potential of the future. And so when death happens, a huge burst of carbon will move out of that structure to signal that there's a new energy source. There's an opportunity for new life to happen. CO2, methane, all of these things are discharged from plants that are dying, humans that are dying. Any organic material, plant, animal, otherwise, is a huge carbon sink. It's a huge reservoir of energy. The building blocks for that energy system is CO2 in the atmosphere. And so for your government to come along and say CO2 is a poison and we're going to start pumping that stuff out of the atmosphere because it's killing the planet, all I can tell you is they are absolutely fundamentally wrong. CO2 is always the building block for the new potential energetics or metabolism for life. We're coming along with chemicals now and overplowing, and we're starving the earth of its opportunity to do carbon cycling. So when the topsoil dies, the earth cannot breathe. So we've basically induced a situation of emphysema where there's plenty of gas to be exchanged. The person with emphysema is breathing, more than I do. They might be breathing 40 times a minute and I breathe 8. But they can't utilize that resource because the lung surface has disappeared. And so as we denude, 97% of our arable soils are now denuded of nutrients and microbiology that can cycle that life. The earth is in a long breath hold. The vast majority of what we call waste on this planet is massive gigatons of potential energy. And all of the CO2 in the atmosphere is the batteries of the future for life on planet Earth. And so we need to reverse our fear, guilt, shame paradigm that industry and governments have programmed in us to guilt us into some sort of effort towards change, and realize that that abstract argument is actually accelerating the death of the planet because we're not realizing that CO2 is not the problem, the lack of lung tissue is the problem. We need to heal the soils of this planet so that Mother Earth will take her first breath. And when she breathes in, folks, it's going to get really beautiful around here.
So, for me, I thought Zach's comments about carbon dioxide were very key to what was the central message that I wanted to bring today. And that is that everything that lives has to breathe. And if it fails to breathe, it doesn't live very long. Right? As Jerry said, cellophane bags all got a warning on them. They don't belong over your head, right? Okay. So we're going to talk about that. And in the context of that, most of us in farming have always had serious agribusiness inputs into our life. They're always trying to figure out how to increase your milk production by another 5%. By now, your cows should be producing about 500%, right, for the breed. They don't seem to ever do that. Your corn variety should be yielding somewhere around 1,000 bushel. Right. All these percentages are really pretty fictitious, right? So there really is my point is there's really no silver bullet. And Jerry has alluded to that in several points in his talk, that really what we have is a system. And the system has multitudinous, diverse relationships, which are all complementary if they're managed correctly. And I think what I've observed over my lifetime is that the master creator designer of the system is a whole lot more capable of making those decisions than I am. So I really need his leadership. You see this dirt on my knees, and that's where I try to stay. I spend probably far too little time on my knees praying, but I do try to stay on the soil in my knees so I can see what's going on. And So that's what we're going to do later on here today, and we'll talk about some of these points again when we're actually looking at the root system. But my challenge is, how about letting our soils do the farming? And I'm kind of, I'm stealing some property from James White from Rutgers, who talks about rhizophagy, who says that we're way too interventionist in our farming practices. We put our nose in places where it really doesn't belong. And we'd be better off if we just took our hands off the wheel and let the organized system, which God put in place, actually run the farm. That's the essence of that. How about letting your soils do some farming? And what we do a lot of times, you know, is shoot ourselves in the foot, right? We planted it too wet. We tilled it too much. We put the herbicide on at the wrong time. Maybe we didn't even need it, or whatever. All kinds of decisions that we make, and they're dangerous. So a soil has to breathe like every other organism. It's essential for the developmental process called rhizophagy. And this rhizophagy is really exciting to me. I mean, the more I learn about it, the more excited I get. Can you all say rhizophagy? It's a new word, right? But you're going to have to learn what it is if you want to understand 70% of what happens to your plant. That's Dr. White's summation of what he thinks the consumption of microbial bodies constitutes in the life stream of a plant. Oh, you thought it was soluble phosphorus, right? That's why you put 624-6 out there in the trench. Yeah, or you thought it was that soluble potash material, or it was the right chelate of zinc, or it was the wrong chelate of whatever. It was something you bought, you put in the system, right, that you thought it was all about, whereas it's only about 30% of what it's about. Instead, it's about 70% on what the microbes are bringing to the plant. One of the interesting things about the exudate question, which I'll digress just a little bit, is there's a phenomenon known as quorum sensing. And it's literally the communication that takes place between the plant and its exudates in the microbial world that surrounds that root. Hello. If you're married, you know that your wife reads you like a book. Right? You don't even have to tell her this or that. She's already figured it out, right? That's sort of the corollary of quorum sensing, all right? You're getting the drift, right? So when this plant needs nitrate nitrogen, it signals, and the food sources that it releases stimulates these nitrogen-fixing nitrosomonas and nitrobacter to create nitrate nitrogen. And this sort of thing that happens early in plant life, why it turns green, stays green, grows quickly. That's based on a lot of nitrate nitrogen. You got something to say there, Paul?
How can you tell?
Yeah. Well, and please interrupt. Please do. Yes.
So you're saying as part of that, those root exudates and sugars, part of that is signals or hormones going out to the bugs in the soil?
Yeah, and it works the other way too. In fact, on my way here, I listened to some guy, obviously from India, talking about root stimulating hormone, auxins, right? And the auxins, I've always wanted the impression that, you know, auxins are created by the plant. Actually, they're not. Auxins, which are key to developing a root system because it sends a signal, hey, let's grow a root. Hey, let's grow a lot of roots, right? Takes a lot of auxins. Well, it's a particular group of bacteria that release the hormones that cause the plant to actually experience auxins. So when you look at root systems out here today, what you're really looking at is the influence of a hormone called auxins. Hello? And that's pretty interesting. We're going to look at those impacts in terms of root system development. And it started with a microbe. And it started with a signal from a plant that said, hey, could use some oxidons here so we could grow some roots, you know? Someday we got to have a root mass. So let's get started. And so there are certain bacteria that are specifically adept at supplying auxins. I think that's extra. I didn't really have that planned. What's very interesting, too, in terms of this carbon thing, is that that plant you're looking at out here, people, is 50% carbon. 50% carbon. And how much money do you spend on the 5% that's mineral? It's less like, wait a minute, what am I investing in here, right? If 50% of the plant is carbon, how much carbon did I need to buy? You know, you guys still buy carbon. I saw you told me what went into the ground in the nitrogen fertilizer. Yeah. Okay, so basically, we know that carbon is in the form of carbon dioxide is absolutely critical, right, to photosynthesis. I was tempted, Jerry, to chime in there and say CO2. You were on oxygen, that's okay. If we don't get oxygen in, we don't create CO2. So that's the way the cookie crumbles. But it's interesting, if you were to ask a greenhouse operator, you know, the importance of CO2, he'd tell you if I didn't put my parts per million in my greenhouse up to 1,000 from 420, I'd go broke. They've learned how important carbon dioxide is, right? But I think we're behind the door yet. We haven't figured this out yet. And as Zach Bush points out, it's absolutely critical to the survival of the planet. At 420, that's a 0.042%. If you're not familiar with parts per million, that's 0.042%. Hello, that's not even a half of 1%. That's not even a hundredth of one. You know, it's just like hydrogen is 0.0005%. And look at the rain it can produce. So it doesn't take a lot of some of these gases to amount to a big deal. This is my point. And CO2 is one of those. So the last thing we want to do is limit CO2. It is merchandise. So here's one of my challenges today. And this stems back to a guy that talked to me 50 years ago, Jerry, from Nebraska. I went to a meeting in central New York, and this guy was a featured speaker to ag consultant professionals in the field. And he showed us a picture of a cornfield that was in severe drought stress. Or at least that's what we all thought. It looked like pineapples, right? You know what that looks like. It's not a very comforting thing to see. And then he went on to his next 2x2 gelatin slide in those good old days with Kodak carousels, you know. And he showed us another slide with him standing there with the wind at his back at the end of the day, and he released a 25-pound cylinder CO2 fire extinguisher into the crop, low on the ground, on the edge of the field. Then he showed us several other slides that he took over the next couple of weeks. And there was this beautiful, healthy, totally relaxed corn right there where that CO2 went, never curled. Again, never showed any kind of drought stress. Maybe it wasn't drought. Maybe it wasn't water after all. Maybe it was CO2. When he supplied it, the stress disappeared. I've never forgotten that. Never forgotten that. When I see corn that's curled up anymore, and I'm tempted to say we could use a rain, I'm tempted to say maybe we could use some more CO2. Well, why would we be deficient of CO2 when it doesn't rain? I'm gonna cramp your style in just a little bit now. We always keep looking to the obvious, don't we? Right? We look at the weather forecast, we look for rain, we look, how long is it going to be? What's the long-range forecast? What's the near-up 5-day forecast? We're always looking to that. But it's actually not the primary source of nutrition from water. If you go back and read your Bible in Genesis chapter 2, verse 5, you'll read there where it says, as of yet, this is the creation record, as of yet there was no green plant in the field. As of yet it had not rained, and there was no man to till the ground. Those three things are in that one verse of Scripture. Now, that condition of no rain existed for about 1,700 years until the time of Noah's flood and God's judgment of sin in the earth. And now it rains, and it's rained ever since. Not the way we'd always like it. But nonetheless, that's what we look to. Well, what was the original system that was the best? What did God create that produced all the fossil fuels that you and I are burning today? Because that's where they came from, right? It was all that vegetation that got buried in the convulsing of the earth under the judgment of God for sin. And we buried all that vegetation, which has today turned into natural gas, crude oil, coal, all kinds of organic substances, right? High in carbon, created by growing plants that grew over 1,700 years without the benefit of a rain event. They obviously accumulated a lot of carbon because when we burn it, we get CO2, right? So what was the source of water? If you keep reading through Genesis, you'll find out that there were fountains of the deep. And today, the vestiges of those are springs, artesian wells, free-flowing wells. Those are actually vestiges of the original system for watering the planet. It didn't need to rain. That was one of Noah's problems, of course, you know. How are you going to float a boat on what? Water? The system was called capillary water. And I think you're absolutely right, Jerry. Water is like right at the top, but it's not the stuff that comes out of the cloud. It's the stuff that comes up from the reservoir of water that came from the cloud, but has to hydrate the whole soil system. That's one of the things we're going to look at in the pit today. And we've seen a change of it in just a few months on how this soil is handling capillary water. Okay? I've probably gone on and totally missed where I am in my slides. Yes, this one, okay. I'm still capillary water. So the key, in my opinion, is that capillary water is going to require a resource. In Noah's day, it was fountains of the deep. There was bodies of water that were actually under the crust of the planet, and the water moved upward, not hindered in any way, and watering the microbiology of the soil, which produced the same kind of CO2 that this microbiology is doing right today. That has not changed one bit since the original design. What has changed, however, is the way in which the soil system itself transports the capillary water. There is the rub. That's why you see the corn curl, and that's why I believe it's not water, it's CO2. CO2 problem created by poor capillary water. All right. So, yes, I've asked you to consider the 2023 corn crop as an example. And I think I may have pointed this out in the winter meeting, I'm not sure. But we produced the second largest corn crop in American history last year, and we did it on record low rainfall. Most of the major corn growing areas of the country were under-supplied with the water necessary to meet the transpiration rate of the crop that came in. So how in blazes did it do it if it was drought stressed? How come we didn't have serious tip back? We did here, didn't we, in some places? And when we dug those roots last year, we kind of observed why, right? The roots were shallow, the soil was bone dry, and there was no evidence of capillary water coming up. What happens when the capillary water comes up is essentially it maintains the vitality and the vigor of the microbial community. And that means that it's going to take oxygen, which hopefully is coming in, turning it into CO2 and releasing it into the crop canopy. And as you pointed out last winter, Jerry, that CO2 never gets above the ear sheath. The plant uses it very quickly, very efficiently in those lower leaves, cycling it back as exudates, right? It's like, hey guys, the primary focus is still the same. Keep the microbial population alive and functioning well at a high level. And all the rest of your troubles, including a lot of insects and including a lot of weeds, including a lot of stuff that we probably haven't even discovered yet, disappear. Just because we have a healthy microbiome because it's constantly getting water. That water is also cooler, rising up out of cool soil, and it manages to do some control of the maximum soil temperature where microbes would shut down. If it gets too hot, they quit. They don't like steaming off and dying. So what they do is they dewater themselves, go into a spore state, and become stagnant. When it cools off, where they can get rehydrated, they wake up and go back to work. Get the idea?
Jerry,
Yeah.
are you going to kind of go into how the soil breathes and how what some of the
Yeah, I am.
practices we do kind of hinder?
Yeah,
Okay.
I will. This is kind of a borrowed slide from Dr. White at Rutgers, where this is an idea of how this rhizology works, how the microbes are actually ingested by the root tip right behind the expanding tip of the root. And that microbe is now basically eroded. The cell wall is eroded by superoxide and ethylene, which is created microbially in the root tip. And then the plant literally selectively farms out the contents of that microbial body that it needs to meet its nutritional requirements. It could be an amino acid, it could be, who knows? But it already knows what's in there because it signaled the microbe to come through the exudate. You getting the idea? It's a really tight fit supply chain system. And maybe this is a time to say this. If we intervene and supply too much of whatever this plant is looking for, let's just talk about phosphorus, okay, for a minute, because we spend a lot of money on phosphorus. If we oversupply available phosphorus, the plant never signals the microbial community to send phosphorus. It's like if you got a long weekend in the automobile industry, they don't keep sending engines and fenders. They say, hey, listen, we're not going to build cars for 3 days, so hold the shipment until we go back to work. So that's what happens. The plant says, hey, hold the phosphorus, guys. Wait a minute. I'm trying to get through this 624-6. Getting the idea? Once the 624-6 is gone, of course, we never trained the new workers. We never hired the guys to go back to work after the vacation, so they don't like to show up.
Does it take a different microbial community to usher the 624-6 into that plant since it's in a synthetic form
No.
versus a natural organic form of phosphorus?
Where the problem
Same.
is that 624-6 is typically an orthophosphate, or even if it's not ortho, it's mostly or partially ortho. So that means it's water soluble. So as the plant root system takes up water, it inadvertently will receive phosphorus. It's no longer in charge of phosphorus supply. It's just been bypassed, right? Because we have mass flow of water that's carrying the phosphorus. And the problem, of course, is that phosphorus doesn't last very long in that form. It starts to react with calcium, magnesium, aluminum, manganese, and other minerals in soil and tie up. So you're really only good for that water soluble for maybe 2 weeks, maybe 3 weeks. By a month, it's about all gone. The impact is spent. And now we don't have anybody showing up for work. That's the problem. It interferes with the quorum sensing right from the beginning when that phosphorus is putting there in the soil and it's water soluble. Okay? All right. We talked a little bit about tillage, and Jerry referred to a very key element there about, you remember the increase in CO2 when the ground's turning black on Route 35? Yeah. What essentially is happening there is it's like putting a patient on an supplemental oxygen in the hospital, and somebody messed up and they turned up the regulator. You know what I'm saying? You supplied way too much oxygen. Yeah, you fried him with too much oxygen. His brain will burn up. And the same thing happens when we incorporate suddenly massive amounts of oxygen into the soil profile. First thing we see is that it releases mountains of CO2. Jerry said 50%, right, Jerry? From 420 to over 600. That's very significant. But what it implies is there's a significant amount of microbial life that perished under an overload of oxygen. Kiss the fungi goodbye. They're done. You pump that much nitrogen in, 78% N2 from the atmosphere, and that's the end of cyanobacteria. All the natural nitrogen fixers fix nitrogen gradually. They don't cotton to being oversupplied. The system is resilient, but it's also fragile. And now you have a major rebuilding process to take place. All right. So this is the mechanics that you were asking about, Paul, wherever you are. Yes, question back there. Sure.
You talked about microbes. How about supplementing the soil with biologicals and different type of fungi? I mean, if you look out there, there's thousands of products. From
Yes, sir.
Some have 2 items in, some have 25 bacteria in.
Some have 8,000.
Can you talk about, either one of you guys, the impact of that? Does it help? Does it hurt?
Well, all the biological products try to supplement what's there. And for the most part, what we're supplementing is pretty inconsequential. And I think the real barometer of success of what you have currently in your soil or what you bring in as a supplementation is the ecosystem or the environment in which you have it. That's where performance comes from. It comes from not just from what the stuff you bought in a jug, it comes from what you already have out there. It was in your manure on a dairy farm. It was in all kinds of things that you use to inoculate the microbial system of your soil. So one of the things I want to emphasize today is that you are a manager of an ecosystem which is impacting the microbial world intimately. It's not just ho-hum, so what? No, it's absolutely critical. I would never myself personally, I'm going to put myself in your shoes. I would never buy a biological product unless I knew I had an ecosystem to put it in that would allow it to perform and to persist. At 6 billion microbes per gram or per teaspoon of soil, just exactly how much supplementation do you think I really need to do? And if I have an ecosystem which is promoting the growth and reproduction of those organisms that I already have, just why would I think I have to resupply? They're all about living. They're all about growing. They're all about multiplying, right? It's an incredible beehive out there of synergistic, complementary relationships. And frankly, if your soil is getting a response out of most of the biological products, you got some serious hometown problems. Problems at home. And we
Jim.
need to change the way in which we're creating managing the ecosystem. Yes.
Sorry about that.
No.
If I've mismanaged my system and I'm transitioning to a system that's going to now support that reintroduction of biology, is that still a good place for products that have species that maybe I have, they have gone down in population because of my mismanagement? Now I've created this opportunity for them to thrive. I want to catch up and introduce those species and then create that environment for them to thrive
Yes,
after. Is that a good option?
I think that is a very viable option. In fact, my clients around the country practice that option because they don't want to delay if they are economically, financially, basically using the same kind of expenditure level to do this. And we often do this just trading dollars, like eliminating phosphorus or largely limiting the amount of phosphorus. And we put into that environment where that seed is phosphorus solubilizing bacteria in an optimal environment where they'll actually perform. That's a good thing to do, and it paid for itself, right? Michael Risey, for example, they don't thrive in a no-till environment. They really don't. Pathogens do. They do really well because it's wet a lot in the upper reaches where these funguses will develop, and disease becomes a serious issue in a no-till environment. We're promoting fungal development, but it's not the right kind of fungi. And so, yes, you can put beneficials in there. And I think this behooves me to mention this. Did that thing change on me already? Yeah. Dr. White, I don't know about you, but when we talk about microbes for years, I conceived of it as a battle between the white hats and the black hats. It was the good guys versus the bad guys. You have that mentality? It's kind of like the Westerns, you know, Roy Rogers and Gene Autry and those guys. You know, it was always the good guys, and again, it's the bad guys, the rustlers, right? And that's not what Dr. White is discovering. What he's discovering is that Fusarium graminearum, which would give you vomitoxin in your corn silage this fall, is actually a good guy. His problem is that his nutritional requirements, which are supposed to be met by the rest of the community in that soil, have not been met. Parts of the community are being lazy, or maybe they don't even exist. Maybe we've done things that have just changed the balance of the microbial community to the point where Fusarium just says, hey, I could use some more nitrogen. Where's all these nitrogen fixers? So what does it do? It goes to the nearest source, invades the root system of a plant, which is excreting ammonium nitrogen, its preferred form. Oh my goodness. And there comes disease. It starts to grow inside the plant where it has no business, and frankly would not even be interested in being if the community had been there to support it. You getting the drift? So there's a lot of good guys out there that turn bad just because of the community they're existing in. So we need to do things to repair the community. And so if you're talking about a biological product, you're probably going to be better off if you're doing this to use multiple species and to make sure that you do everything you can to optimize the environment for things like mycorrhizae. Okay? Good question. Great question. Yeah. Mark? Yeah, Mike is coming. There you go.
The soil is living. We got all this microbiome out there, and to his point, once you inoculate your soil with microbes that you believe are the best microbes, you've got to keep them fed.
Yeah.
Correct? What are we
Yeah.
going to do to feed them during the year to keep everything healthy? And number two, what are we going to stop the bad habits for killing them off?
Well, yeah. So we need some alternative ways to make a seed bed, don't we? Yeah. And the other point you were asking in your question, I've lost it.
How are we going to feed these
Feed 'em.
microbes?
Yes, feed them. A community actually supplies its own food resources. It's based around carbon, and we know that's why sequestering carbon is really important. And by carbon, I'm not talking about a piece of corn stalk, right? That's not what we're talking about. That's just plant material. That's not usable carbon for the most part. There are some species which will work on it. Some of the fungi will help to decompose it, but we're talking about a carbon resource that is active, that is soluble carbon, that microbes can use to create cell walls and all kinds of things that are food sources. They're called saprophytes. That's their job, is to create food sources out of decomposing plant material. And so that's the key. And root exudates are another one of those food sources, obviously. The carbohydrate, Jerry said, right? It's a sugar. There are different kinds of sugars, and there are proteins and all kinds of things in those exudates. And we're going to take a look at here in just a second about some of those exudates and how they've influenced soil nitrogen, for example. So that help, Mark? Yeah. Does that answer it? Okay. Well, what this bar graph shows is something I showed at the meeting in January. I did this study in 1984. And at that time, what I did was analyze every inch of soil that had had water sitting on it up to 2 feet deep for 4 days, that all of a sudden, when we poked a little piece of steel in the ground 6 inches, the water disappeared. And I was provoked to do this simple agronomy 101 exercise called a mechanical assay. I separated sand, silt, and clay, determined the percentage of each one in each inch down to 7 inches. Because mechanically, I knew we had done something by penetrating that soil with just this little piece of steel that today is evolved into what we call the CurseBuster. And so when I did this, I discovered that there was a moving amount of silt particles, and it had accumulated in the 5th inch somewhere. It was probably 20 thousandths of an inch thick. It only had to be thick enough, it only had to block enough macropores where air and water pass each other to be able to stop the air from coming up, which prevented the water from going down. And it sat there like that for 4 days on an alfalfa field that wasn't even 2 years old yet. This was in northern New York on a beautiful silt loam, creek bottom. And my client was very upset because I had told him to break the plow sole, which he did very faithfully. And he stayed off it with a disc. He didn't put a sweep on it. He did everything very minimally with a little Danish tine and his brilliant seeder. Sounds perfect, right? No compaction left. Water ought to move forever. And it didn't even move for 2 years. And I didn't know what to do. I told Donald, I said, I don't know what to do, Donnie. I'll help you drag hose and carry gas and we'll pump it off, you know. The creek was on the other side of the field. He said, no, I got a better idea. He said, take that New Zealand thing you brought around last fall. We thought was a joke. And he said, when we run it through that water, I think we'll drain it. And he was right. It was gone in hours. Fortunately, I had the county agent live just down that road, Jerry. And he drove by while we were making fools of ourselves. He came back at 7 o'clock that night. It was 4 in the afternoon. He was going home to have supper. He came back at 7 with this little Chevy Love pickup, 2-wheel drive, and summer tires. He said, I drove all over the entire 35-acre field. There wasn't water anywhere. So at that point, I knew there was something about this. And Klaus Martins, I don't know if any of you know him. He's an organic grower of 50 years in upstate New York. And he wrote to me one time, and he said, well, the phenomenon is known as microerosion. And it was discovered in the late 1930s by a German soil scientist. And the reason he called it probably microerosion is because he discovered it with a microscope. And he saw the erosion was movement of silt under the influence of water. And if you look at stream bed formation, if you ever took a geology course, which I had the distinct pleasure of doing, you understand that stream bed formation actually is a horizontal, visible representation of this exact same phenomenon. Moving silt horizontally, forming various soil types along creeks and rivers. That happens when water runs down, carrying silt with it. And when the water slows down, it drops the silt. And it'll do it every time. And when you get to around 20 or 25 inches of precipitation or snow melt combined, or irrigation water, you got the problem back. And if you want to know the basis for it, go to Genesis 8:21, where it's rained now, and Noah and the boys and girls are all up on Mount Ararat, and God speaks to him, and he says, I'll never again curse the ground for your sake. This is the anatomy of that curse. And until we learn to deal with it constructively, we'll continue to destroy the ecosystem of soil. It in itself is destructive. You know that. When you don't get water movement in your soil, things are going backwards, ain't they? And that's what happens here. Water sits on top of this density layer because the air underneath it cannot exchange with it. Ever try to fill one of these bottles with a garden hose? You can stand there all day long, point water at that bottle until you get the air out of the bottle. The water ain't going in. Correct? I see a few heads bobbing. It's really simple physics. Very simple. You've experienced it. Okay. So essentially, what I'm suggesting to you today is that in order to create the ecosystem, we have to have water behavior, which Jerry's talked about, and we have to have gas exchange. We have to be able to move air up and down, changing CO2 and oxygen. And the only way you do that is when water behaves itself. And that's one of the things we're going to look at today, is how water is behaving itself between where the accelerator is run and where the CurseBuster ran last October. October? Thereabouts. And that was the total tillage and the total difference. And you see this, I don't know if you can see the stain on my knees or not, but I was over where the accelerator ran this morning, and I didn't have anything sticking to my knees, on my pants. But I moved over where the CurseBuster ran, Jerry. I started getting, there it is. It's sticking to my pants because it's wet. There's a dramatic change already in the hydrology of the soil as a result of basically addressing this phenomenon known as micro-erosion.
So you're saying it was wet because of capillary action moving up?
Well, in this case, we are actually seeing water percolation. Okay? If we didn't percolate it and store it, then we couldn't get it to come back up as capillary water. All right? We got to create our own fountains of the deep here. And that means we got to store the water efficiently and effectively so that we can get it back. All right?
But that's why your knees are wet, because of the capillary movement coming up where you weren't seeing that on the other side.
Well, it was just the soil itself was so much wetter that I was chipping off the wall.
Okay.
Yeah, where Dave had made the pit for us out there. Yeah, it was just damper. The subsoil was wet. Yeah. Okay. This is going to be virtually impossible to read, so I'm not even going to try to bore you with it. This is a little bit more readable, and it tends toward where I want to go. But I want to point out a couple of examples that we see when the ecosystem is working better. In this case, I'm looking at nitrogen. And of course, all the textbooks tell us that nitrate nitrogen moves with water. So when water percolates down through soil, it takes nitrate nitrogen with it. And if you go on my website, you can see this in a video, and it's very much more legible than it's here on this screen today. But the fact of the matter is, I'll just cut to the chase. We're changing the entire paradigm because we've changed the microbial community and its functioning. And instead, this slide actually is even better. This is a 2019 sample, and nitrate nitrogen was 30% in the top 7, 35 in the second 7, and 35 in the third 7. In other words, when I sample nitrogen on your farm, I'd make 3 cores, 0 to 7, 7 to 14, 14 to 21. And I analyze for nitrate and ammonium nitrogen in those 3 zones. And now what I'm doing, obviously, is discriminating and trying to identify what's happening with movement of nitrogen in the profile. Where am I getting changes? And 3 years later, this same field, sampled in 22, had 46% of the nitrate nitrogen located in the top 7 inches. The next 7 inches was down to 24, and the bottom 7 was down to 30. And what I didn't show you is that the amine nitrogen is actually increasing in the bottom 7, which is very nice, and we won't digress into all that, but anyway, when you've got microbiology that's really working and soils are really breathing, a lot of things that we believed were true are no longer true. It changes fundamentally. And here's a fundamental change from Saskatchewan. This guy was growing spring wheat, 50, 55 bushel spring wheat, and putting out about 110 to 130 pounds of N to generate that crop. And last year, he grew 500 acres of spring wheat and 500 acres of durum with no supplemental N. And this was the reason. His soil test showed that in the top 7 inches alone, he had 68 pounds of nitrate nitrogen in December. After the crop was off, that soil had continued to fix nitrogen. And so when spring came, he said, I'm going to just wing it. And believe it or not, no supplemental nitrogen was some of the highest protein. It was 14.5% protein on that wheat and durum. And it was beautiful stuff. It was, just great color. Test weight, 64 pounds, and normal yields. And he bought not 1 pound of N. This is a 6-year phenomenon, okay? Soils that have been in that 14 to 17-inch rainfall period. So in a couple of years, he could have this density phenomenon, this microerosion phenomenon kicking in, but he continues to perforate it without mixing and turning the soil. He just restores normal gas exchange and water movement. And this is the kind of result you get. Nitrogen fixers love to live on the top of your farm. When you don't leave them there, they're unemployed. You just ruined the workplace. You getting the idea? All right. Well, this is a little story from Wisconsin and a dairy that I just had to tell because Mark Wagner would have loved to have been here. This is Mark out in December running his 30-foot CurseBuster behind a tracked deer. And it was a field of hay that had been down 6 years. First cutting was always a lot of shepherd's purse in it. They didn't even put it in the bunker for the cows. They just put it over in the heifer arena, you know. Sure, there was a little alfalfa there, but the grasses were totally taking over. The second cutting was abysmal, if it was any. And so he said, you know, we're going to just, the whole deal was, we're going to cut the first cutting and then we're going to plant it to corn. We've had it, right? It's over. And so he went out in December and said, well, if, you know, if I kill it, I kill it. You know, I haven't really lost much, right? Well, that's the way a lot of guys approach running this technology in alfalfa. You know, they'll use it on the worst fields so that if it dies, it's no big loss. So that's exactly Mark's mentality. I don't know if you can see the picture or not. It's pretty dark. But that was his first cutting this year, and he said it was one of the best fields they cut. The shepherd's purse was totally gone. There wasn't a single plant on 70 acres. He said the grasses were so covered up you had to part the canopy of the alfalfa in order to see that there were any grasses left. And he said the second cutting, the grasses have totally quit. And it's just pure alfalfa stand again. That was a stand 6 years old, ready for, you know, the plow, ready to kill. And that's the kind of things that happen once you start to restore the soil's ability to manage air and water. The microbial community says, well, it's about time you took the straitjacket off. And that's kind of what it's like. You know, you don't really need to cuff a villain. You know, you don't need to put him in handcuffs. All you got to do is put him in a straitjacket. You can leave him right there because he ain't going to run very far before he runs out of air, right? That's the whole idea. And that's what happens over time with our soils. They become like your man with emphysema that Zach Bush talked about. He breathes a lot, but he's not breathing very much. Not much air change. All right. And that's a picture of some corn that we grow over in Wisconsin and Spencer near Marshfield, which has got a very appropriate name because it's all marshes. And Jerry's shaking his head. He knows what I'm talking about. And if you look at those root systems, which I apologize, it's not any brighter to see, you'll see that there's just massive amounts of hair roots there, massive amounts of soil adhering to those roots. That's what Jerry's talking about. We're going to see some of that today. And eventually, in a fairly short period of time, you'll see the kinds of auxin producers that generate this kind of root branching. The other thing which is real interesting, and I'm going to digress one last time, and that is that, remember I talked about rhizophagy and superoxide and eroding the cell wall so the plant can take out the contents it needs? You were all here for that, right? Okay. The hair root system that develops is actually a response to having digested microbes. Oh, you thought it was for absorption. So did I. I thought that was great, but it's not. It's like that porta potty back there. If you drink or you eat, you got to go to the bathroom. And if this plant is successful at actually eating a microbe, it has to get rid of the spent microbe. You know, it served its purpose, and we need to get rid of it. so it grows a colon. It has a bowel movement. If you see those hair roots, that plant's going to the bathroom. It's going to the bathroom because it ate. It's pretty easy, isn't it? Kind of basic. So when you see a plant like this with all this hair root, that tells you there's a lot of eating going on. This guy farms organically and grows 200 bushel corn, like falling out of bed. And when you see that root system, and when you saw it in real life, you really kind of like, oh my goodness. How do you generate something like that? Well, there's no interferences from chemicals because he's organic, right? One of the things about the 23 crop that I want to emphasize and why water management and gas exchange are so critical is the 2023 crop year featured a dry start. Right, Jerry? It was pivotal. It was absolutely critical to see the outcome. Because when it's too wet, too long, the soil is actually microbially going backwards. It's generating compounds that somehow it's got to resolve before it's going to actually produce a healthy crop. That's what we got this year. Too wet, too long, too much ethylene being produced in soil. It's going to produce a shorter corn. That's not where ethylene belongs. Remember, ethylene belongs inside the root, where it decomposes the cell wall. When it's outside the root, it develops a meristematic tissue that doesn't like to grow a normal cell length. It's literally a plant growth regulator. You can buy it in a jug. It's called Ethifon. It's synthetic ethylene. And people that grow various vegetable crops and high-value crops use ethylene all the time because they want to shorten the crop. They need to tighten it up. Okay? So part of the success of 23 was the fact that we didn't have to recover a backward condition. We just proceeded smoothly, seamlessly. Great root systems, great gas exchange, no bad compounds to take care of, to deal with. That's why we were successful. And so the importance of dealing with this microerosion problem on a year like this is obvious, isn't it? If you're doing a better job of percolating water, then you're going to do a better job of exchanging gases and minimizing these bad compounds like ethylene that form in the soil. Right? Okay. Jerry, what do you want to add?
One thing.
Okay. Get a mic over to that man.
Okay.
And we're going to have questions.
Yeah, I just wanted to comment. You talk about the microerosion that's within the soil profile.
Yes, sir.
It only takes 1,000th of an inch of crust
Oh, yes.
at the surface to block gas exchange. And so when we talk about microerosion, we're talking about really thin layers. And if we have that crust on top of the surface, not only within the soil profile, but on top of it, we limit everything. And we just see that again and again. So it's a matter of how do we maintain that structure of that soil in its integrity to allow all of this. And I don't know that we can overemphasize just how critical gas exchange is between the soil and the atmosphere and within the soil itself.
Yeah, I mean, it's, well, I'm going to share this anyway. I hope you're not offended. But I saw this gentleman, he was in a church, and he was explaining the name of God. The name of God is Yahweh. And if you say the name, you realize you're not using your lips, and you're not using your tongue, but it's uninterrupted air in and air out. Yahweh. And his same pattern is what we're observing is so powerful in the life of the soil. Is it a surprise? I mean, he is the creator. Would you agree? He put this thing together. It should be his character being manifested in our lives, right? In Noah's day, it says they didn't swim, the length of an Olympic pool to beat on the door. It says, in fact, the breath of life was taken out of everything that walked and crawled on the earth. They never felt the ground come up to meet them. When the breath of life goes, you're dead. You could be standing at the time, but that's it. When the breath of life is gone. So what we're really all about is reintroducing breath and breathing to soil. You getting the idea? So it's powerful. That's the point. It's powerful. Like you said, we really don't grasp the full magnitude of the significance of gas exchange. We probably don't appreciate it ourselves in our own lives unless we put a bag over our head. Then we appreciate it pretty quick, right? Soils are telling us the same message. We just got to figure out how to read the mail. Question? Skeeter? No? Yes.
So what's the bottom line? I mean, why don't we just keep on doing what we're doing and doing synthetic fertilizer and covering up with fungicide? And I mean, why change?
Well, I think you're going to run out of money, number one. Yeah. The commodity prices right now are already making people rethink a few things, right? And I think it's a good cycle because it does make us rethink some stuff. And it's amazing. Jerry alluded to this, how it impacts insect pests, how it impacts pathogenic bacteria and fungi and organisms that can be bad behaviors. And it changes the whole system. It changes the attitude of everything. Last year, I was at Wagner's farm scouting at silking time. You know what for, right? Long-term corn silage round and rootworm, yes. And beetle-feeding silks, yes.
Hey, Jim, quick question.
Yeah.
How much more profitable are your customers that's been using this biological way and really focused on this oxygen and this carbon cycling?
Maybe one of the best ways is to say that the Wagner dairy was averaging 200 bushel corn between silage, high moisture, aerolage, and so forth, all bottled down. Last year they averaged 250. And they used no fungicide. This year they're using a half rate of insecticide in the trench and a fungus called metarrhizium to control soft-shelled larvae forms, cutworms, rootworms. On 2,500 acres, we cannot find a gooseneck plant anywhere. Yesterday, he was out scouting for beetles. He saw 2 on a 100-acre field.
Something that I've observed going to your customers' farms or people we've been working with in this biological journey is if you're a dairyman or if you're beef or no matter what kind of livestock you're doing, it doesn't smell as much.
Well, the reality of it is, Mark, and I appreciate you bringing this up. If we go back to the phenomenon of rhizophagy and the technology known as endophytes, all right, these are bacteria and fungi that move into a plant and they literally reproduce and grow inside in the intercellular spaces of the entire plant. Those come into your bunker silo in that plant material. And if those are beneficial microbes in soil that nurtured a healthy plant, guess what? They're still in the bunker. And they're still in the TMR that goes in front of your cow. So the manure that comes out of that cow is different than what it was when you were doing fungicides and insecticides and essentially sterilizing a plant. Dr. White has said now that a typical seed will have up to 90,000 colonizing bacteria and fungal forms on and inside the seed. 90,000. That's a legacy and a half. You forget the DNA. forget what we bred into the plant with pollination and inbreds and all that stuff. Think about what the inheritance is from the microbiology that wound up in the plant that's now in the seed. And that's why he's a big promoter of save your seed. If you got a healthy soil making healthy seed, use it. I wish it worked with corn so much more, but anyway.
And I'll share a quick story when we're talking about carbon cycling and the importance of oxygen to the microbiome. I got a customer in southern Ohio, 160 acre farm, 160 acre field. He says, I cannot get over 150 bushel on this farm.
Yeah.
What's wrong? And I went in there and I knew what the answer was, but you got to test to have a convincing story.
Yeah.
So I used a company out of North Carolina called Rise Bio. I really like what they do. And it was just an easy problem of there's not enough oxygen there to support the microbiome. So therefore, a lot of the microbiome's been retarded or killed off. So we inoculated it with the right microbes and got him to pull back to 160 units of nitrogen rather than 220. And this corn's going to be the best corn he's ever had out of all his 7,000 acres.
All that nitrogen you see, just like the phosphorus example I was citing before, interfering with quorum sensing, that nitrogen dose like that is discouraging all the nitrogen fixers from showing up to work. They've been replaced, right? We've heard about replacement, right? Anyways, I have another farm in the glacial till area in central eastern Illinois. It's where the glacier stopped. It's not that nice, deep, black stuff that you hear about and think about from Illinois. It's quite the contrary. And they've historically been in that 195, 200 range for a long time. And in their second year of embracing the microbial approach and using a CurseBuster, they went to 230. Nice corn year. Everybody figured that's the last time we'll see that. Last year, they experienced 3 rainfall events, had great capillary water. Anytime you were on the farm, didn't matter how long it was, you scratch a 1/4 inch down and there's moisture. Didn't matter. They went from 1st of April to the 3rd week in July without a drop. And 3rd week of July, they collected 3, next 3, and the next week 3 more. Total of 9 inches, and they broke 230 again. And so it's not the precipitation event which is really driving the result. It's the CO2 being hydrated by capillary water that drives the result. Water's very much there, but it's not necessarily the rain event source of water that's making the significant difference.
I've got a seed question too, and this is an observation. This may be pointed towards Paul and his affiliation with Pioneer. We've all kind of been studying the uses of brace roots, and we always think that's a good thing. Brace roots, another 2 or 3 collars high. It's not a good thing. It is a response of that plant saying something is plugged there. I need to have whatever nutrient is, and they shoot off another set of brace roots. Mm-hmm. Is that your thinking too?
Well, yes, the vascular bundle, which filters the water which is going into the plant, is plugging, usually with excess iron or aluminum or manganese or some other mineral or mineral compound, which is being taken up because soils are chemically out of balance. It's usually a function of mass flow, what's in the water resource, what's soluble, versus the microbes would never do something like that. I mean, the microbes are pure. They're absolutely pristine sources, in my opinion. They don't make mistakes, right? They're just answering the call. But if you have vascular bundles that are plugging, of course, the plant responds by setting another set of roots at the next node up, trying to bypass the block. It's just like any good plumber, right? He puts in a new piece of pipe where the old one's shot. So, yeah, it's not a good situation. And interestingly, Mark, in 2023, I could hardly find a plant with a brace root on it in how many states I was in. This year, with that wet start, I got brace roots where I haven't seen them in years. It's just unbelievable. They're back, and it's been very disappointing in some respects, but it tells me that there were bad things happening when that plant was young. Yeah. Paul.
This
ahead.
is a good place to break. I think it'd be really interesting to get out to the root pit, unless you had some other things you want to talk about first there.
Oh, yeah, we will. Yeah. And it's a good time to visit the porta potty. If there's any line over there, there's a really good bathroom, too, inside. Just the door on the right down the hall. First door on the right. All cleaned up, guys. Don't worry. I took care of it. All right. Yes, we'll adjourn. Grab another water because it'll probably feel really good in just a little while out there now. And any last question or challenge or complaint? Anything for the good of the order? All right, good. We're dismissed and we'll reconvene at the hole in the ground.
So from where the flag is to the right or towards Jim, we were on the CurseBuster once last fall after bed pack manure, and to the left would be a Krause Excelerator vertical till disc, and then this spring we worked again with the accelerator over here, but where we had hit the CurseBuster in the fall the one time, we just left it, and that didn't get worked at all. So that's kind of what we're comparing here. I would assume on a normal year we would have CurseBusted or made a tillage pass before planting here.
Certainly, probably in the early adoption
Yeah.
period, a lot of farmers benefit from a second pass.
Yeah.
Over time, that tends to go away, and one pass in the fall is sufficient.
Yeah.
You were saying something about the sequence of spreading the pen pack manure. You ran the Krause Excelerator over all of it, didn't you?
Yeah,
After
so
you'd spread it?
we had the manure spread on the field, and the whole field was worked with the accelerator to incorporate the bed pack manure, and then we were able to get our hands on a CurseBuster, so we came and reworked this area with the CurseBuster, and that pulled a lot of the bed pack straw and corn stalks back up on top of the ground, where the accelerator probably had 50% incorporation. After we ran the CurseBuster, it probably pulled 90% of it back up to the top.
Yeah, and that's very characteristic. It's mostly the rotary harrow in the back, which loves to comb material out of the profile and dump plant material back on the surface. So you can see that, I think, if you look on both sides, you'll see quite a bit more bare soil over here where the accelerator.
You can still see it really obvious.
And it would be two passes of the accelerator, really.
Yep.
One in the fall, one in the spring.
Yep.
So, and we like to see that because we know Jerry Hatfield likes to have a blanket, right, Jerry, to control that raindrop impact on the surface. So we like to see plant material left on the surface.
Anything else you want to add?
Anything you want to add, Paul?
No, I didn't. Oh, you did. Okay. Oh, yeah.
All right, so we did a infiltration test this morning while talks were going on. For those of you not familiar with it, basically you pound a 6-inch ring into the soil. These two are right about 5 inches in, so we have about an inch left. And then you put an inch of water and time how long it takes to go in. So that's kind of a simple test to figure out how fast water can infiltrate into your soil. So to start off, for the first inch on the CurseBuster, it took 2 minutes and 3 seconds, and on the accelerator, it took 3 minutes and 5 seconds. The second inch was 8 minutes and 34 seconds on the CurseBuster, and then 9 minutes and 26 seconds on the accelerator side. I then kept going. I did on inch number 3. I had to go back and get more water, and it went down faster than I could get back here, so I don't have data for that one. And then inch number 4, whoops, come on, phone. Inch number 4, it took 18 minutes and 47 seconds on the CurseBuster side and 16 minutes 26 seconds on the accelerator side. So we've done, and then I did another inch after that during lunch. So
Okay. And the way we did this is a little bit unique in that normally it's more like a 2-inch event. And the problem with that as an infiltration test, of course, is that if the soil is capable of holding the 2 inches of water and it's above the density layer or any barrier to percolation deeper, you'd never know until you put more water on. And my point has been with people over the years, when a glass is full, it overflows. So infiltration is really only as good as percolation. Once you've exceeded what you can perk, you will have zero infiltration.
Do you have any no-till testing, or are you testing no-till accelerator?
not in this area, no. I don't know if Jim.
No. But if we understand the nature of this micro-erosion phenomenon and know that no-till is not addressing that density layer, and what I know you're alluding to here, Mark, he's always the devil's advocate. You got to read between the lines, and they aren't very far apart. Yes. Yeah. Yeah. He packs a 36-inch baseball bat, okay? So beware. So basically, you have that density layer that's going to limit percolation. So you'll look fine on no-till as long as you don't get too much rain at a time. But when you do, and the density layer is stopping good, effective percolation, although the infiltration looks great with limited rainfall, it falls out of bed when you get too much. So that's the point of breaking that density layer and rendering it harmless in terms of preventing gas and water exchange in the entire plow layer. Which, when you get 5-6 inches of rain, you could use the whole plow layer, right? It's a little over the top, but we see it, right?
What I was getting at in my area in Ohio with the Maumee Bay watershed and algae bloom.
Hold that, Mark, right there. Start over.
What I was getting to, what I was wanting to talk about was in my area in Ohio, around Findlay, where they have the Maumee Bay and algae blooms being a problem, and the USDA and NRCS office is helping us make it worse by adopting no-till the wrong way. And I think what we're studying today with the CurseBuster will kind of solve that problem.
Yeah.
Nate, do you want to talk a little bit about what you like to see for water infiltration?
So that's basically how fast you can get an inch of rain into your soil. So the higher, the better, I guess is a short way of saying it. In this area, I've seen everywhere from like a low of 2 minutes to a high of an hour. So obviously, as we get these thunderstorms during the summer, the where it's taking an hour, you're not going to get most of that water. I can't remember right off the top of my head, but I think in the more severe thunderstorms, we can get about an inch in 5 minutes. Is average for this area. When we got a summer storm, obviously, if you get a slow rain, it's gonna be a lot different. But just kind of give you a reference idea of maybe what you should be shooting for in that first and second minute.
Mm-hmm. And you recorded it was faster for the first 2 inches events over here.
Yep.
How much do you think that was attributable? Anybody wants to comment because of the amount of residue that was left? Was that left inside your ring?
Yeah. So I didn't clean any of the residue out. Just pound the rings in, and then I do pack around the inside of the ring because as it goes in, it vibrates a little bit and opens up. So it could run down the inside. So that's the only part I mess with is right on the inside of the ring. All the residue is left.
Right.
Where it should be.
Questions, comments, concerns? Jerry, what do you think?
Well, I was going to make a comment that impact of residue with raindrop energy is different than the impact of residue when you do an infiltration ring. The infiltration ring, you're flooding that whole environment. You're not letting the raindrop energy really impact it. so it's moving down by gravity pretty quick. So those are comparing apples and oranges. So anytime that you've got a residue layer on top of that raindrop hits it and then it slowly goes down through. So the more we can keep that residue layer intact, the higher we'll maintain that infiltration rate. And it'll even probably increase it a little bit above the flooded piece of that as well. Just because it's even at 1 inch in 5 minutes with a rainstorm is still running a lot slower into that soil than is dumping that equivalent of the 1 inch of water in a ring. So just be cautious about interpreting some of these numbers from that standpoint.
And one of the other things that I asked Nate to do was to make sure that the ring was down to an already located density layer, which has developed on the accelerator side, because I didn't want the water to escape around the bottom of the ring and miss the problem, right? So that's why we used more than 2 inches, and that's why we drove the ring in 5, because we wanted to make sure we were into that density layer, which could diagnose the percolation issue. If you've ever seen the old Great Plains ad where they were selling turbo tills, they put the ring in about 2 inches. And so it looked really, really good. But of course the water was diffusing underneath the ring. So it really wasn't evaluating the situation very accurately. But it sold turbo tills.
Yeah.
Yep.
There's sales literature and then there's reality.
Yeah. Okay. You good? Paul, you good?
Yeah. So what are we seeing here?
Well, all right.
Before you jump down in that pit, remember Paul asked the question this morning about maintaining optimum efficiency of that plant and what it means. And if you just feel this corn leaf, it'd actually be better if it wasn't so cloudy. It's actually cooler than air. You go out in your fields, anytime that plant is cooler than air, it's transpiring at its maximum rate. And what we want is we want the maximum rate of transpiration because that's the maximum rate of photosynthesis. So anytime that plant leaf starts getting close to air temperature or above air temperature, it's stressed. And it's no longer as efficient at getting creating photosynthesis, creating that exudates that go down. So maintaining these temperature cooler than leaf temperature during the whole day that's what we use as an indication of lack of stress or an optimum plant growth.
I've heard a lot of, like when you get over 85-degree air temps, the plant starts to shut down and go into respiration mode. Is that kind of what you're talking about?
Not necessarily 85. Plant can go above 85 leaf temperature. Starts going into stress at that point because if it's 85, just give an example of this. If the air temperature is 85 and we had a bright sunny day, we'd probably have leaf temperatures that were maybe 75. They'd be 10 degrees cooler than air. And it starts getting up above 85. Now we start really being stressed because we might be a little cooler than air, but it just cannot keep that much water moving through the plant. And so it really becomes a water dynamic process in terms of how much energy can we move. And water is energy going this way. CO2 is energy going that way. It's all an energy-driven process. And so we can keep that. This corn plant, during a really clean summer day, the CO2 levels in this part of the canopy will be about 290 parts per million. Remember I mentioned that the atmosphere is about 400 and something? 290 is where C4 plants reach their balance point. And if you can keep stirring that air and keep that CO2 level above 290 parts per million, we got optimum as well. So not only are we thinking about water, but thinking about how do we get that CO2 replenished into that canopy. So that's that balance that we start thinking about in terms of optimum productivity, creating sugars, all these different pieces. I just wish it was bright and sunny. You could really see. Of course, it'd be hotter than heck if it was bright and sunny out here too. So,
What's the ambient right now? Anybody know?
What do we say?
What do you
Was
got
it
on your weather?
About 80 degrees, isn't it?
I think it's a little over 80. I'm not sure.
I don't know.
It's interesting, Jerry. I was just scanning over here while you were explaining all this. And the upper leaves in the canopy are around 74.
Mm-hmm.
And further down in the canopy, they're running 76, 77, 78.
Yeah.
They're hotter down inside the canopy. What do you got for ambient?
82.
82.
8 degrees cooler than air.
Yeah. And you can turn around and pitch the plant. You can actually feel the leaves are cool to the touch.
Oh, yeah.
Definitely. Yeah.
You
This
don't, you don't need an infrared thermometer. Just go out and feel them. You're fine.
You may not know the number, but you can tell.
As long you know when it's cooler than air. That's that.
Yeah. This appreciate Dave loaning this to me today. I left mine at home where my son likes to use it. But this infrared has been very indispensable in terms of my understanding of gas exchange, because air, of course, can move heat energy in it. We use it in our houses, right, and in our shops to distribute the heat from the furnace or the wood stove or whatever we've got there providing heat. If we didn't put a fan in that shop, all you'd have is a hot spot around the stove, right? You'd still be freezing over on the other side of the shop. And so it is that we can have all kinds of solar-produced soil temperature on the surface of a field and not really change the soil temperature at all, because we didn't move the heat down. How many realize it, right? You remember that general science, all heat rises, cold sinks, right? It's no different out here in the field. Just because you made it brown, right, which some guys are still pretty enamored. We got to have a dark soil surface, right, in order to build soil temperature. I can take you to a lot of farms where there's a lot of dark soil in the spring, and it's still cold. Because it didn't move the air, which contained the temperature, the BTUs, down into the soil profile. So this thing has been very indispensable in diagnosing that. Often I'll be called in the neighborhood area and said, hey, Jim, what are soil temperatures running? And if I'm checking curse-busted ground, it's usually 10 to 12 degrees warmer at 2 inches. But the most unique part is that when I find it at 55 or 60 at 2 inches, it'll be the same temperature at 7 to 8. There's no gradient left. It's gone. Whereas if it's a no-till environment or something that was minimally tilled in the fall, and there's been no real improvement in efficiency of gas exchange or water movement, you'll have a gradient. If it's 55 at 2, it'll be 50 at 3. And it'll go on down into the low 40s by the time you get down to that 7-inch profile. You got one of my clients in real trouble with his eldest brother. His eldest brother always wants to be planted first. There's 4 guys farm together, and oldest brother gets to be number 1, and number 2 brother actually is the one that runs the planter. And we'd been studying these soil temperatures, and he realized that he had 65 degrees on the 15th of April, 8 inches deep. And so his thinking on that was that instead of planting shallow, which we've always done when we plant early, I'm going to plant deep. And when, of course, we had 3 weeks of winter after that, his brother was quite concerned, so he's been out there digging seed, and the first day he found a seed at 3 inches, everything collapsed, right? The phone, I would not have wanted to have been in the younger brother's boots that day, because I'm sure he got chewed really well. Everybody knows when it's cold and early, you plant shallower. When it gets later, you plant deeper. You did exactly the wrong thing. What he was thinking is pretty obvious, right? He was putting the seed down where there was energy and staying power to maintain the energy, instead of being concerned about how fast can I recover the soil temperature for the seed I dropped 3 weeks ago. Didn't do anything. Instead, he said, let's just put it in the heat and keep it there, because there's plenty of heat. And above it, the soil temperatures did get down into the low 40s and the high 30s in those 3 weeks. But the seed never left the high 40s and low 50s. So that meant microbiology continued to work in the seed. All the microbial activity was still functioning as normal around that kernel of corn at 3 inches. So it does change a few things. sometimes it can make some strained relationships. Okay? The other thing that we discovered using this little device was just this summer, where I usually go out on this same farm and I dig a 24-inch long trench with a tile spade. I go down 21, 22 inches or more, and I measure root activity to those depths, because I want to know if I'm testing nitrogen in 3 layers to 21, I want to know who's coming for supper. I could have nitrogen there, but if I don't have any rooting activity, I'm not going to put that in my calculations for available nitrogen because nobody's showing up. Got the idea? Well, this year I happened to grab this, put it in my back pocket, and I shot it at the ground 24 inches deep, and it read 81 degrees. I was like, 81? Normal soil temp here is like 55. What are we doing with 81 degrees? And it kept coming back no matter how I scanned it. 81 degrees. And it cooled off, actually, a little bit more before it went to the surface. So we went across the road where they'd run a high-speed disc for 2 or 3 years. And we did a shallower trench, only 14 inches, and we measured the temperature. It was 74. And it cooled off under that. So it's very interesting. You can actually diagnose how thorough your gas exchange and water movement is, because those are both sources of increasing soil temperature. Right? It's going to be air movement and water movement that tempers that 55 degrees. And in this case, at 2 feet, we're running 81 degrees, optimal for microbiological activity. And we could see it, and we saw it here too. We're seeing mycorrhizal fungi colonization, the production of a biofilm on the root system at this, 3 foot? We were seeing it at 3 feet. So that's just great. That just adds productivity to the soil, adds character and health. And so one of the things that I want you all to take your turn down in here, actually, as many of you as want to come down in, I'm going to kind of give you a flyover, but what we saw is basically related to the phenomenon that I discussed. I don't know when I discussed it with your people, Paul. We talked about root system physiology and how that by 40 days or thereabouts, the root system has established its boundaries. In other words, you kind of staked out your territory. It's like what's on your deed, right? Your land deed. And you're going to work within those boundaries to run your farm. And you do that first, right? I mean, you're not going to start farming ground until you've got the deed for it or some other official arrangement to know where you're working, right? So that's what the plant does. It's a little like the way our country evolved. We fought a war against Spain. We bartered with Britain for the northern border. We claimed the Pacific. And we basically set up boundaries, and there was no downtown Minneapolis. There was no railroads. There was nothing but a trail that supported movement of military forces, and we established national boundaries. The plant does this in 40 days or so, and then it decides how many rows of kernels it's going to put on the ear. And the embryos begin to form at that point. And there could be multiple embryos on several nodes on the plant. And eventually one of them will emerge, or maybe 2, as capable of actually bringing forth fruit. After that 40-day period, through till pollination, which is just around the corner, this root system now fills in within the boundaries. It's called the establishment phase. And I owe this to Al Trouse from the National Tillage Lab at Auburn for teaching me this many, many years ago. And so then once pollen drops, all new rooting activity ceases. And what you've got at that point is what you're going to have. That's what you're going to finish the crop with. So early development, establishing boundaries, is really the first and very critical step. And you can pick those roots out for their size. You can also pick them out from where they originate, up near the seed. And there's 5 major seminal roots that are the home base for this. And we saw here in the accelerator that we had good root development here. In fact, these soils had a lot of encouragement from what Joseph, were you telling me, or yeah, Joseph was telling me about the biological program and stuff that was put into the root zone at planting time, which stimulated a lot of auxin production. Remember we talked about auxins this morning? And that auxin production has resulted in a very nice development up here in the top. I'm apologizing to you guys behind me. If you want to come around, that's fine. But you can see up here in the surface, and we'll get the shovel on here in a minute, and we'll see what the whole plant looks like. But you can see we've got some good major development here. The problem we have is that every root tip that originates, that emanates from a root initiation, only actually grows and expands for 72 hours. And it wears out. That whole rhizophagy function thing, the big rush is 72 hours. And then that root tip essentially matures, kind of like when you turned 18, 19 years old and you didn't get any taller. After that, we grow in other ways. Suck it in, Jim. Okay. But because of that 3-day interval, we can see here that there is some kind of inhibition, because down here, we really don't see very many main roots. The boundary setting phase was truncated here. And if you think about it as a 3-day event, you realize that if there's some kind of physical barrier here, or if there's some kind of chemical interference, which interferes with root growth, then we're not going to get as far. And there's kind of symptoms of this here, isn't it, Jerry? Very definitely. And it shows up with the concentration of root systems that would have developed within the boundary. The problem is we never got the boundary by 40 days. So therefore, we didn't develop much bigger root systems. Did I confuse you all? I don't see too many people nodding.
Restate that a little differently in saying that we had a boundary at 40 days, but it wasn't the maximum boundary that plant is capable of.
Absolutely. Yes, it was imposed environmentally.
Yeah.
Precisely.
So
what we wanted to do was see, is there a difference? Did we actually successfully increase the boundary setting phase? And the way we diagnose that is by looking at the secondary branching that took place between 40 days and pollination. And we found roots this morning. They're still growing. They've still got expanding root tips on them at 3 feet. Well, that means that there was a boundary set somewhere below 3 feet. It would take another backhoe run up the row and deeper to find how much more deeply those root systems have actually gone. An undisturbed pit like this will actually tell you, because the next morning you will find little wet spots on the top where the root systems are bringing water up from below our pit. It's pretty exciting when you see it. Anyway, so this is typical. Here's this dividing line. And you can see down here, it's not as vivid as it was, but when it was nice and fresh, it was pretty easy to see we had major amounts of root development down here. Where's our rock, Jerry? There it is. I just lost my microphone. I have no idea how to get it back. That's fine, yeah. Is it still working?
Still working.
It is.
Before I fall in.
All right.
I told you you should have stayed.
Did you say that? Stapled it. At least some goop. All right. Jerry, I can't find that root in that pile of dirt.
Yeah, it's probably dried out in
Anyway, there was some really nice clusters of root systems that were down here at 3 feet. And so what that tells you implicitly is that we had major roots that made it down in excess of 3 feet, because we're filling within that boundary. Okay? There it goes again. It's up in my eyeballs. All right. Anyway, what else do I need to talk about here? there's an example right here. Oh, the other thing I do, which you guys can do this, it's easy to do. I was telling you about the soil temperature thing. When we dig to look at root systems and make evaluations about nitrogen utilization, what we do is dig between the corn row. Everybody knows there's roots in the corn row. The question is, did they make it to the middle of the row? And how deep did they get? So I dig 2 feet deep in the middle of the row at 40 days. And if I'm all the way out there and 2 feet deep, I know that any nitrogen resource I've got basically in 21 inches of soil, any place on the farm, is effective in growing my crop. And that's how I base my side dress recommendations. I test for it, and then I evaluate the root system and go from there.
Jim, when some people test for nitrogen and they go 2 and 3 feet deep, what are they gaining there? When people are talking about testing for nitrogen and they go 2 and 3 feet deep, what are we really talking about? What kind of results are we going to get there?
Well, I guess what I would have to say is that if you don't have root systems there, you don't really learn much. Right? I mean, I kind of figured if you're going to fix dinner, you need to know how many people are coming to eat. Right? So before I go out and buy a bunch of groceries, I want to know what I got in the pantry. So I'm going to count heads. I'm going to look at the root system, see how deep it is, how thorough it is across the whole field. And then I'll make a decision about how much of the nitrogen I found is actually useful. The reality of it is, a lot of nitrate leaching that we see is actually after the crop is done taking up nitrogen. And soil is not done giving you nitrates. But there's nobody there to gather it because the plant is already done. So the reason I think we see that data that I showed you that you couldn't see, and we see nitrates accumulating in the top 7 inches instead of leaching, is because we have microbiological activity that's functioning at a high level in decomposition of the pre-existing root masses. And so therefore it's tied up. It's trying to correct and hold that CN ratio in that 10 to 11, 12 to 1, and that means it needs some N because there's a bunch of carbon in that root system. The bigger the root system, the more the carbon you got, the more N you need. And so these nitrates that we're seeing are basically being held there from metabolic decomposition processes. Good for groundwater, good for the pocketbook. Okay? What I thought I'd do is see if I couldn't, where am I here? Where's my little flag? Right there, okay. I'm going to see if I can get us a plant. This one's good, right here. I'm going to try to get out as much of this as I can. And this is much more typical of what I used to do. But we'll see if it's instructive at all today. Where's one of your strong men? Come down here. Somebody catch this thing, Scott. Can you catch it? I can't do both. All right. All right. Can you set it right over there? All right. And we'll do the same thing over here on the CurseBuster too. Scott, yeah. All right. I'm just going to leave it like this, and I want you to look at it, and I think you'll be able to see the impact of a density layer which is formed up here, and you can see the character of those roots. Let's come over here. And we'll do a similar kind of a thing. If I can miss your rocks. That's a doozy right there. Yeah. All right. If you can move that over toward Jerry, I don't know how you're going to do. You can't feel this, but I can feel the primary root mass snapping. Could you hear it?
I can hear it.
You could hear it? I didn't hear that over here. That's the difference in boundaries. Okay. All right. And we can turn that around a little bit, and when people come down in the pit, what you'll see here is you'll still see the remnants of this density layer. It's still here. You remember we talked about the density layer of silt still in place, but it's rendered ineffective because we've basically made a hole. And we've stretched it, we've fractured it in such a way that it no longer limits gas and water movement. It's still there, but it's not effective anymore. We were having that talk at the table, right?
Still there temporarily.
Yeah. And the root systems will get rid of it. They'll, boy, Jerry, I'll borrow that or Paul. So basically what we have to do is deal with it in such a way that we eliminate the negative associated with water movement when it's too slow and the lack of gas exchange. There you go. So one of the things that we have observed that is a direct result of effectively dealing with this phenomenon of microerosion is that you have so many more primary roots and secondary branches that are operating at a much greater depth that we are no longer using rippers on plow sole. It's happened a lot over certainly the last 10 years. And to some degree it was happening before that with this kind of tine technology. But it's really happened big time once we introduced the CurseBuster and we made more of this destruction of the density layer obvious to the root systems in the plants. And what they'll do is they will literally approach the plow sole and they will grow into it as much as 2 inches a year. A farm that I referred to briefly in Illinois on that glacial till. They had a 4.5-inch layer that was so thick with a small point on a penetrometer, I couldn't get through it. It took 2 of those boys to get through it. That's how we knew how thick it was. And last fall, the third harvest, second at corn and 1 beans, no cover cropping, and that density layer was totally gone. The needle never left the green zone on that penetrometer. And we've done nothing to address it with steel. And if there's one other overriding message I want to leave, and that is there is not a piece of steel ever been built, conceived, that I know of anywhere which actually creates soil structure. You're chuckling, Jerry.
That's a tagline I use is that you can't till your way to soil health. Sorry, you can't buy a green piece of equipment and makes it work. I do want to make a comment because he's talking about the different layers. And I just did a glance in that system versus this system. Just look at the prevalence of brace roots. Remember you talking about brace roots this morning? Many more brace roots on that side up on the second node. Rarely you see, maybe on the first node here. I mean, so reinforce your point that you made this morning.
So anyway, the point I want to make about root system activity is that only root systems and their degradation or decomposition is what creates soil, in the old word, tilth, right? That's what I knew it as growing up until soil health came along. And by that time, I had clients in northern New York that had achieved wonderful soil health, and we didn't know what it was called, except tilth. And so those root systems, as they decompose, they actually are the ones that create the aggregation, that produce the structure that resists compaction, that expedites water percolation, infiltration. All the stuff we're looking for does depend on us growing a root system. And that's best a steel is going to do. Best steel is going to do is give you a better root system. a more accurate representation of the genetics of the plant to generate roots. And that's the foundation for which the microbiology will operate. If you have a small root mass, you're not going to support a lot of microbiology on that root mass. If you have a massive root mass and it's deep and it's wide, it's going to support a much bigger population. Getting the idea?
Jim, we got some dairies here, obviously, and
They got to milk.
well but they got to get rid of manure and tankers and compaction and silage boxes and
It's a little early to milk.
Well, I don't know. I'm not. I'm just
A little bit.
So, like, if they've been running the CurseBuster, do you still see if they've been running manure tanks or silage boxes that they still have to rip once in a while?
Yes, we were having a small conversation a while ago. I think maybe it was with you, brother. We were talking about compaction issues associated with traffic and harvesting and spreading manures and so forth. And my point was that when you put rebar in a piece of concrete and you do it right, you can park a loaded semi on there and it won't break. But if you put that same amount of rebar in and you don't put it in correctly, you'll break the concrete every time because the rebar was not installed correctly. So root systems which grow in an abnormal direction, like parallel with the field surface, that's bad rebar. It don't work. But if you grow it at an angle, like it should normally be, a normal architecture, the resilience, the resistance to loading and compaction forces is huge. Absolutely huge. It doesn't take long. I mean, you can see the difference in the depth. You can see the difference in between the rows. And you're very welcome to come out here, take the shovel, and dig in between the corn rows and observe the difference. I hope you will, because that's the foundation for resistance to compaction. And when it does appear to be compacted, Paul, it's really shallow. You've seen the pictures. You've all seen the pictures, right? Where it goes down 2.5 feet, with a 8,000, 8,500 gallon tanker. You don't see that when you got the right rebar. It's within reach easily of 5 to 6 inches. After that, there's nothing there. Okay? That's the way it is. It's all about architecture. It's all about root system response to what we do. And we don't do it all. There was a long time I heard people preaching against sweeps and discs, and the compaction issues that they generate. But the reality of it is the originator of a problem of compaction was the one who said it's going to rain from now on. And that authored the whole idea of microerosion and silt movement. That's the origination of a backward soil. And you didn't have anything to do with it except that it was for your sake. That's what he said in that context. He did it for our sake. So I think it was maybe that in Noah's day there was a no-tillers club. He said, I'm going to show you that doesn't work. How many no-tillers we got here? Yeah. So, yeah. I guess I've pretty well said what I thought I wanted to say.
Did you want to show a little bit more about how the moisture profile?
Oh, yes. Thank you for doing that, because that was something Jerry and I saw this morning. And I said something about it during my talk, how my knees got muddy when I got over on this side of the pit. It's because we had more moisture down in the profile, deep. And, of course, if it stops raining, now we have a resource deep in the profile and no resistance in terms of a density layer that's going to stop capillary water from coming up and hydrating the microbiome of the soil. So it kind of, the traffic runs both ways on this road, You've got to put it in the ground or you're not going to be able to get it back up. And the same density layers that limit how much water you can get down in the ground are the same ones that are going to limit the amount of capillary water you can bring back up. And if you want to come down here and poke around in that sidewall, you'll see to the south of that flag, that subsoil is totally different. Dave, you saw it with the hole. You want to tell us about it? You got it?
When I started digging on this end, it was coming out of the hole, like a clump of dirt and stuff. And when I got over there and I got to the lowest bucket down, It just kind of fell out and fell apart, as I got down there. You could see it was drier.
So, Mother always, Mother always used to tell me Rome was not built in a day. So what we're seeing here is actually something which has transpired since last October. We were more effective at moving water deeper in the profile since October. And we can feel it right now in that subsoil moisture, which put that mud on my knees. And over there, we did not get that experience, and therefore the reservoir is dry. You with me? So it's an investment in the future of the crop you want to grow down the road, nearly a year later, to be able to effectively store water. Okay. Thanks, Dave. Question? Yeah, the question was concerning the height difference in the corn where the CurseBuster ran. It is perceptibly shorter. And I don't know what you have to say about that, Jerry, but my experience is that if a root system on a plant has a greater measure of difficulty at establishing a normal architecture, its response is to get taller. I'm not sure why that is. I have no explanation, and maybe you do, Jerry?
No. I mean, the feedbacks between roots and shoots are a little bit a mystery so far in terms of height, in terms same number of leaves on each plant. It's just a difference in internode length, and a lot of it's probably due to auxins within that soil that are being generated that says, you don't have to grow to be 10 feet tall. You can do your job at 7.5. So, put on some roots, will you? Yeah, just put on some roots. I'm better off putting energy into root systems than I am trying to go capture more light. So, again, one of those things in physiology we really don't have a good clue on yet. We need more field physiologists out making some of these measurements rather than just breeding more corn. So.
There was one other thought I had today that came up out of the 2023 corn crop experience, Jerry, and that was that how does 9 inches of precipitation get you 250 bushel corn? We talked about the CO2 thing, right, and the fact that the plant has got 50% carbon in it. And so CO2 is really probably the major determinant, which, as Jerry pointed out this morning, is a direct result of having good gas exchange, or in other words, the soil being able to receive fresh oxygen supplies, simultaneously nitrogen in the form of N2. But the point is, probably 35 years ago, Jerry, I think it was Iowa State, did a study on how corn accesses moisture above ground out of water vapor. Are you familiar with that study? What was the outcome of that?
The upshot on that whole thing was that there's a lot of water vapor, because now it goes back to the same thing with leaf temperatures and air temperatures and things like this, is that you can pull water out of the air. And stomate's open, things move in, you keep that stomate hydrated and things like this. So, but don't rely on that to create that 250 bushel corn crop. It's a matter of survival more than growth. And so C4 plants have become very adept at working within their environment. Just contrast that to cactus in the desert, which only open their stomates at night when it's cool. C3 plants, entirely different mechanism. They just run full bore until they collapse. Corn plants are built to survive. And
and I was recalling, I don't know how you guys noticed it here in Minnesota, but certainly in the states where I was, nighttime temperatures were extraordinarily moderate.
Yeah.
And so the amount of water that could be held in the nighttime air was considerably higher than our normal 90-degree nights or 85-degree nights. And so there were a lot of things that really favored more utilization of the limited amount of rainfall and precipitation that we got.
Well, and the other piece of that in 23 was also the cooler nighttime temperatures, as you mentioned, because the cooler the nighttime temperatures is the less respiration we have. So all this photosynthesis going on during the day, if it's really hot at night, we can respire it all back out. So we have little left for growth. The cooler we keep those nighttime temperatures in late July, August into early September, the higher the yields as well. We see when we have really high nighttime temperatures is we limit grain filling. We limit the whole growth process of each kernel out there. So we had a collision of events in 23 that made it all work. But the bottom line was that plant is very efficient in taking water out of good soil. And that's
Which, as you know, my parting glance would be, which is what fires up the microbial activity to make the CO2 really come to work. It fills out what's missing in the atmosphere. Yes.
So I guess we started with soil health with Jerry. What is soil health? And I guess thinking back to maybe when Czech first started doing the minimum till is when we were all getting ramped up with that question, and our response was, we need to make this home. I need to stop tilling or tilling as much and destroying the home of the biology that's going to provide life for this plant. And I guess in evaluating minimum till versus conventional till, which is, I guess, the two we have in our area the most, what we've been finding the last few years on maybe just disking ground, we have that layer at whatever 5-6 inches, and below that it's hard, slaty looking. You can break it up, it's in compressed layers. Everything below that root zone, we're only farming the top 6 inches. So in those areas we've gone through, and it's like, well, we need to rip this. We need to break up those layers. So we're getting down deep with an inline ripper, trying to break that back up, alleviate that problem. But we're going right back to conventional tillage and reinserting those layers. So then we've moved towards this minimum till where we're going to lightly incorporate our manure or our residue in the top few inches, try not to disturb much biology below that, and then still finding that these fields are awfully hard to probe through for soil sampling. They're hard to, I'm digging and I'm finding wormholes down deep, but they're not very large in diameter. The soil still feels compressed, and I still feel the need to rip. And I guess we've noticed nice probing soil sampling wise in the fall on minimum till fields that have been ripped, say after year 3 to 5. I guess I just want your thoughts on that type of system, because that's kind of where we've come to with trying to create that good home for the biology. And now you bring in this idea of gas exchange and water and that movement, and that can be kind of new for some of us. So where do we go from here? What are some practicals?
Jerry's taking a vie on me.
That's a Jim question. No, I think you bring up an interesting dilemma in saying, all right, how do we balance tillage? How do we balance the biology? And how do we look at that soil structure? And the first thing I guess I would ask is saying, what is that, going back to the root boundary, what does that root system look like on that plant 40 days after emergence? Have we really captured that profile? Because roots will expand into, if we can go to mid-row at about a 30-degree angle, that's what we've discovered as we dug roots and everything. That's the optimum angle out there. Is how do we bring all that together? You don't really want to continue to over-till the soil, but how do you judiciously use tillage to create a seed bed that endures over time? And that's really what you're asking, is saying, I don't want to look at it 1 year. I want to say, here where I'm at in 20, 21, 22, 23, 24, and now going into 25, what's my best management practices I can put in there? And it really is, I think, looking at that root system, looking at microbial activity, and looking at the penetrometer is a good measure of saying, do I have that hard layer out there? But, you can see the slaking in this, but with that hole that root was able to get through it. So now we still have to start looking and saying we need to dig a few roots between the rows, see what they look like in terms of extension, look at it in terms of what that plant looks like late in the season in terms of efficiency and in preserving those lower leaves. I always look at the lower leaves and telling me how well is that plant really functioning over time, because if it's firing up to the ear leaf, that plant's scrambling. It's sort of like the brace root analogy and everything else, because if I'm robbing those lower leaves, that plant says, I'm in the process of survival and creating seed, and I'm going to do it at any cost, and lower leaves are the bank. And so I think it's not really an answer to your question of saying, what, do this, do that, but I think we've got to make some observations of saying, are we headed in the right direction? Are we changing aggregates? Are we changing soil respiration? And are we really cultivating a microbial system that works for us, and let that be expressed back in terms of what the plant's doing as well?
So the Jim part of the answer, right?
Yeah.
I think the battery's dead, maybe.
No,
Could it be?
you're good.
You still hear me? All right. The Jim part of the answer is buy a CurseBuster. Let's cut to the chase, right? Well, I'll tell you why. And it's really just physics. It really is physics. It's the physical aspect of the soil paradigm. If we don't address the curse, which is produced by movement of silt particles, then it's going to ensue from there to be anaerobic every time it rains. Soils need to go anaerobic briefly. They don't need to be there handling water long term. They need to restore normal oxygen intake and CO2 production. The way in which the tool does it is really kind of fascinating, and it's not anything that I created, okay? All I've done is try to preserve what the inventor did in the 1970s in New Zealand. Very simple. He fasted and prayed for 1.5 years before it worked right. And he was happy that his pastures weren't going to die and the root systems were going to blow, just totally changed. When the tine is entering, it's actually displacing soil on one side of the tine, which then is pushing sideways forward of the center of the rotating member. And the result of that is stress cracks, which are continuous, consistent in their density. Root systems are finicky when it comes to density. I've discovered, I think, that it's about 25% change in soil density and a root system stops. And it doesn't matter if it's harder or softer. It just doesn't like change. So if you produce something that is creating a density layer that's harder, it could be 20 thousandths of an inch thick. As far as that root system is concerned, it goes all the way to the center of the earth. And it's got to think about this. It ain't got time to think. It's only got 72 hours to get it done. Right? So if it spends 2 days thinking about what do I do, or going sideways on the top of a 10 thousandths of an inch smear zone, we just lost a lot of potential. So there's nothing about this tillage technology that works horizontally. That's the main event. It's working vertically, and it's producing fracture lines which run in such a way at a continuous density that root systems go, hey, man, this is 6 lanes around Chicago. Let's go. And they are on their way. That's why it's incredible how much root mass they generate. It's because the density is so friendly. There's no bumps and there's no speed bumps. Getting the idea? So that's the Jim answer, okay? We want to avoid anything that represents a density change which will limit the ability of root systems to put dry matter in the ground. And provide housing, of course, for the microbial community, which is going to be the basis for real advancing soil health. That's the Jim answer. Write a check. No.
Jim, I'll just be you almost lost him there. I'll be the devil's advocate for just a second. Like, there's a lot of vertical tools on the market. What are you talking about?
Yeah, well, yeah, it's the first time I knew that a disc could be called vertical. You know, I think most all of you see through this, don't you? I mean, it's like, really? All of a sudden, vertical has become a disc, or a disc has become vertical? It's kind of an oxymoron, right? And the root systems show it. Yeah, we went across the field there at Kratzer's in Indiana, and where the vertical till speed disc was used for 3 years or so. We found root systems in the top 4 inches.
But how come so this situation where these guys been running the accelerator, you guys usually run it at 0 to 5, aren't you, or 1 degree? Why are they still seeing the issues?
Well, a lot of times these tools will actually get down to where the density layer actually is, and they will not be able to penetrate it. They literally walk on top of it. It's not that they've necessarily created one, it's just that they've not resolved the issue of one. They just don't run deep enough. And they're typically not designed to run deep enough, unfortunately. So we don't have the problem resolved.
So
That you may not have even created it yourself.
But if you say inline rip that and then continue with
Well, typically, unless it's a Ross inversion, right, where you see virtually no surface disturbance whatsoever, no movement of soils vertically being rearranged, most other of these rippers, disc rippers especially, require you to do a considerable amount of secondary tillage, which is historically done in a horizontal plane. So automatically, you have introduced that horizontal barrier, that density change, where those root systems will never realize that you did a rip. They never got there to appreciate it because they were busy dealing in their 72 hours with something you did at 3 inches. I work with some folks down in South Carolina that have been running a ripper-planter combo, which is very popular in the coastal plain. They've done it for 40 years. And they finally called me up and said, you know, after 40 years, we've decided this don't work. Okay. They're persuaded after 40 years. Wonderful people. They bought a CurseBuster, and they said, now the root system that we have under our corn with basically no real serious initiation like you guys did here to promote auxins, they've got a root system. They said, we can't hold it in our hand anymore. I said, you like that root mass? That's pretty good. Oh, yeah, we've had root systems for 40 years. You can hold in the palm of your hand. I'm like, oh. So things are already changing in terms of the microbial community and its performance to produce a root mass. Cow manure is a great deal with this thing. Guys are just opening up the ground so that it infiltrates quickly, and they surface apply the manure. No more injection. And that's what's producing that data I showed you this morning that you couldn't see on the screen. It starts with surface application where we have the most biologically active soil to deal with what is largely a carbon source. You put it out there, you think you're doing NP and K. Well, you are, but you're doing a whole lot of carbon. Hello? So you need to put it where you've got some cheap nitrogen and the microbes that know how to make it to go with that carbon. When you bury that manure, you put it in a place that's basically naturally nitrogen starved.
And oxygen starved.
And oxygen starved. Yes. Yep. The engine, the fire engine, right? Just had to throw that in there for Skeeter.
So we're after 2 o'clock here, so we don't want to keep you too long. But yeah, definitely you're welcome to dig around in the pit, and or we can work with you too on that.